Um, Anyways, I'm sure we'll get back on track. So I'm Brennan O'Leary. I run a seed biochemistry lab at AAFC Saskatoon, and I'm talking today about a project called Towards a Full Understanding of Canola Germination and Seedling Performance. This is a project led by Sally Vail, but as she's presenting another activity in the next talk, I'll take the lead on giving this presentation. Other collaborators on this project are Rob Duncan, Rob Golden, Janitha Wanasundera, Steve Robinson, Dwayne Hegedus, Isabel Parkin, Rong Zhu, Joanne Ernest, and Leon Kochian. So a lot of things must go right to get timely and successful stand establishment with spring canola on the prairies. Achieving this across fields and seasons is a continual challenge for producers. Poor stand establishment can be explained by numerous factors, including tillage and seeding practices, seed bed quality, moisture, temperature, insect pressure, as well as seed quality, which is the focus here. On average, only 50 to 60% of canola seeds put in the ground actually survive to become a plant. So as a seed biologist, on its face, this is a pretty terrible sounding statistic. And it also has associated economic costs. Seeds are often the most expensive input into the canola crop, and we hate to see many of those seeds going essentially wasted. And there's also a risk of production loss due to insufficient plant density. Unfortunately, plant establishment is a complex problem as a seedling must run a gauntlet of obstacles to reach establishment, which is typically the 3 to 4 true leaf stage. But this process all starts with the seed. The germination and vigor of the seed lot are major factors underlying emergence potential, and germination begins when the seed imbibes water and ends when the radicle emerges from the seed coat. And this process is very sensitive to soil conditions. A given seed type will have a threshold level of moisture and temperature below which it will not germinate and at which it germinates only sporadically. Furthermore, under highly adverse soil conditions, the high secondary dormancy potential of cultivars grown in Canada may be contributing to poor germination and volunteer canola in subsequent years. Once germinated, the seedling vigor of the cultivar or how quickly the plant grows and develops is another major factor contributing to plant emergence. So emergence is noted when you can see the cotyledons through the soil, and this often gives the first direct indication of how things are going with that crop. And from the Canola Council website, I can quote that a canola stand that emerges uniformly, achieving 5 to 8 plants per square foot, not only withstands flea beetle attacks better, but also facilitates more efficient fungicide application and harvesting, ultimately leading to higher yield and increased profits. So achieving rapid uniform emergence is the goal here, and this has positive knock-on effects that smooth the way to better establishment and ultimately yield. So seed germination and seedling vigor of the seed lot underpin the seed-related emergence potential. But here in the prairies and elsewhere, we always have to contend with the elements. And focusing on temperature, which is a major factor, generally soil temperatures below 10 degrees result in progressively poorer rates of germination. And this spreads the germination and emergence out over time, which is undesirable. Canola seeded into cool soils also has lower levels of seedling survival. This is at least partly due to the fact that the clock starts running on seed treatments as soon as the seeds take in water. So a slow emergence means that there'll be a smaller window of protection on those seedlings from flea beetle once they get above ground. So all these detrimental effects of seeding into cold soil must be weighed against the demonstrated yield advantage to seeding early. So the main message of this study is that this trade-off between seeding early and lower emergence, it can be tilted further in the grower's favor if we can identify genotypes and traits linked to the ability to germinate and emerge rapidly in cold soil. So breeding and selection for improved seed and seedling vigor in canola is possible, but it's highly multifaceted. There are many traits involved. So seed vigor includes traits such as seed longevity, dormancy potential, germination speed, seedling growth, early stress tolerance, and others. So to unwrap this web of germination, seed vigor, and plant establishment related traits, it's critical to understand which factors are controlled either by seed genetics or the environment. And that's either the maternal environment where the seeds were produced or the stand establishment environment, mainly talking about soil conditions. That is, we need to know the genotypic contribution or heritability of seed establishment traits. So to achieve this, we're turning to the canola nested association mapping population, or NAM collection. And this is a germplasm and genomic resource designed to study complex traits of spring Brassica napus. This collection was created and managed by Isabel Park and Steve Robinson and Sally Vail at AAFC Saskatoon. And others here today could certainly explain this collection better than I can, but suffice it to say, it's a great source of genetic variation in canola upon which to screen for trait variation. So you have 50 very diverse founder lines that are all crossed to a single reference line. And following several rounds of single seed descent, the result is 50 families of recombinant inbred lines, summing to about 2,500 reels in total. And there are levels of accompanying genotype data for each line. One of the growing advantages that we hope to further improve with the NAM collection is that it's been widely deployed across various research programs and research groups and studies. And this has generated really a wealth of phenotypic data related to these lines. And one of the explicit goals of this study is to generate a centralized NAM trait database. This obviously will have intrinsic value in the database itself, and it will improve the usefulness and the power of using the nested association mapping if you have access to a lot of other trait data. I'll briefly go through the objectives of the project, which I organized really into 3 groups. The first objective is to obtain direct phenotypic measurements of seed and seedling vigor by different experiments. We've got experiments done under controlled conditions where we'll measure seed germination under cold temperature and also plant vigor measurements looking at seedling growth rates. And previous and ongoing field trials throughout the course of this project are going to look at emergence traits for NAM reel lines. Alongside those direct measurements of seed and seedling vigor, we expect that we'll be able to find traits, other traits that help explain variation in seedling vigor in the NAM population. And so we'll be collecting additional phenotypic data on these related traits. So example, one would be soluble carbohydrates or sugars, variation in their levels in the canola seeds. We expect that these might explain some of the difference in seed vigor. And so we'll screen those across the parts of the NAM collection. Very same thing for the protein profile of the seeds, particularly differences in the seed storage proteins. We'll look to see whether they influence the ability of seeds to emerge vigorously. And lastly, imaging seeds for microstructure with a focus on the seed coat. Differences in the seed coat may affect germination ability, and we'll be using light and synchrotron microscopy to accomplish this. And the last objective, which I mentioned in the previous slide, is to generate a seed library database of all the observed traits for the NAM collection. And we'll combine this with the genotypic data for all these lines, and this will allow us to bring to bear, the best in computer modeling and statistical analysis to understand relationships among traits and really to fully explain, or explain as best we can with available data, what is controlling differences in seed vigor. So I'm going to highlight some new technology that we're using in this project to collect germination measurements. And so typically, traditionally, germination measurements for seeds are done on this blue filter paper where the emergence of the radicle, so the germination event, is clearly visible against this dark background. And usually this would be counted manually, sort of once a day, and you can also have time-lapsed imaging to capture the rate of the radical elongation as a measurement of seed vigor. So we're going to take a different approach, and we're going to rely on high throughput respiration measurements using this robot sensor to measure germination. The assay works by putting individual seeds in these tubes, and the tubes are sealed, and they essentially act as an oxygen sensor. And as the seed grows, it consumes oxygen, and from the trace of the oxygen consumption over time, we can identify exactly when the seed has germinated without having to count them every day. And so what this kind of looks like, these are traces of oxygen consumption over time, and when the radicle breaks through the seed coat, you get an obvious acceleration of the oxygen consumption or the respiration rate, and that indicates exactly when germination has occurred. So it's got a higher level of accuracy than the traditional method of manual counting, which again was only done every couple of hours. But the other major advantage of this technique is that it can discriminate between non-germinating and dead seeds. Dead seeds don't respire at all, whereas non-germinating or dormant seeds have a low baseline level of respiratory activity, which allows us to distinguish, that which are dead and versus dormant. And that's an important distinction that's difficult to make only by visual examination. So now I'll present two examples of developing data sets. First, these are germinations assays of the NAM founder lines using the respiration machine I just showed. And so we've conducted these assays on seed lots sourced from multiple locations. We're at 4 different locations now, and we've tested at 2 temperatures. And what's very encouraging is we see a tremendous spread in various germination metrics across the NAM founder lines. And this data here shows that basically after 10 days at 5 degrees, some founder lines have not germinated at all, and some have reached 100% germination. So that's pretty good extent of variation right there, phenotypic variation. And that's very positive. A negative is that there's high levels of environmental variation from the maternal environment, the seed production environment. This is very clear, and this will be something that we have to deal with on moving forward. However, some of the lines are consistent across environments in being either very good or very bad germinators at low temperatures. And one bit of good luck is that NAM 0, which is the reference line to which all other of the founder lines are crossed to make the NAM collection, it's a consistently poor germinator. So if we look at crosses between NAM 0 and consistently good germinators like NAM 74 or 75, we can expect to observe the desired phenotypic spread in these families that allow us to search out QTLs explaining germination potential. There are also extensive ongoing field trials of select subsets of the NAM population, and during these field trials, a suite of measurements are collected throughout the season with a focus on imaging tools to track emergence, establishment, and progression. For example, along with manual counts, we are utilizing the AAFC Pro tractor, which is an adapted plot sprayer equipped with 4 to 6 GoPro cameras on time-lapse mode, which collect pictures of the plots while the tractor drives along through the pathways. And you do this multiple times a season, and then the images are rapidly analyzed digitally. And this is just a summary of data from a 2023 early seeded nursery in Saskatoon. It contains plots of 16 NAM families. So each of the box plots is a family, and each family is composed of 28 to 58 rills. And the metric here is just it's just a metric of emergence, really. But what's important is the data shows a good range of emergence phenotype within most of the rill families. Again, that's going to allow us to hopefully detect QTLs moving forward. So wrap up quickly, we want to identify genotypes and traits associated with seed vigor. And this will lead us to genetic strategies to improve spring canola stand establishment in the prairies. Environmental effects will remain a major obstacle and something we have to contend with moving forward, both seed production environment and the seed germination environment. And last but definitely not least, the production of the NAM collection traits database will have, I believe, great intrinsic value moving forward. With that, I'll just thank all the collaborators who helped out with this project. Thank you.
So thanks for the opportunity to present. My name is Sally Vail. I'm a canola breeder, Agriculture and Agri-Food Canada in Saskatoon, and I work largely in germplasm enhancement trait development, so upstream from the commercial industry. And I'm going to present today on activity 6, getting more from less. And it's a pretty wide-ranging project with a lot of different collaborators. I think they're all co-PIs because they're all leading different objectives within the project. But the overarching goal is to identify canola germplasm and traits that will hopefully reduce the environmental footprint of canola. And this is through the reduction of nitrous oxide emissions, but also looking at strategies to increase the carbon sequestration potential. So one of the major objectives that I'm going to be talking quite a bit about today is on nitrogen use efficiency. That's why I've underlined it in the title there. Major objective is field studies and genomic studies on variation in nitrogen use efficiency or NUE related traits, as well as plant architecture. And also co-leading this objective is Isabel Parkin and Steve Robinson. There is a couple sub-objectives which are looking at NUE, plant architecture, and root system architecture under controlled conditions. So Isabel Parkin and Steve Robinson here in Saskatoon have a really interesting lysimeter plant array phenotyping system where they're looking at natural variation for these traits, and also looking at some gene-edited lines with some interesting root phenotypes. And also Raju Solakanahali, a physiologist here in Saskatoon, is using a steady state system and taking a very different look at the NUE picture. He's looking at largely ammonium tolerance in modern hybrids and working with some really interesting physiological models. And then on the soil side, we're working with Jennifer Town from Ag Canada, as well as Melissa Arcan and Bobby Helgeson from the University of Saskatchewan. And a couple different objectives. Jennifer and Bobby are looking specifically at lines with contrasting root system architecture and looking at the impact on the microbial community in the rhizosphere, and with a focus on nitrous oxide production and consumption and the nitrogen cycling. And Bobby and Melissa are also looking at the carbon produced from canola and tracking it through the rotation to see how much of that carbon ends up as soil organic carbon versus greenhouse gas emissions. And also looking at soil versus fertilizer uptake between lines. Okay. Oh, here we go. Okay, so the big question, why? So there's, we're probably all aware that there's both economic as well as environmental considerations as to why we need to look at nitrogen use efficiency. The chart on the left is showing what the input costs for producing canola are, pulled from the crop planning guide, and nitrogen fertilizers anywhere from 15 to 20% of the variable costs that go into producing canola. So if we can figure out some mechanism or ways to improve nitrogen use efficiency, there could be economic benefit to growers. But then on the environmental side, we're probably all familiar that our government has some really rigorous targets in terms of greenhouse gas emissions, especially from fertilizers. With reduction of 30% by 2030. And I should mention, when you dig into this documentation, a lot of it is targeted around agronomic practices, which I think we'll hear about in this session as well. But this project's looking at longer term goals in terms of breeding targets and ways that we can improve cultivars for nitrogen use efficiency. So the question that this slide is asking is, what is the best breeding target to improve nitrogen use efficiency? And this conversation really started at the beginning of the previous round of funding in CAP, where our team sat down and took a look, or had a series of brainstorming sessions where we were saying, well, what is the best target within the plant that we can look at to improve nitrogen use efficiency? And we quickly realized this wasn't really a very easy question to answer. And this is just because of the complexity of the end cycle. It's not only the plants and the plant genetics, it's the soil, it's the environment, it's the atmosphere, it's the continuum. So it's a really complex system. And then in addition to that, there's a plethora of ways in which nitrogen use efficiency is measured. So looking through the literature, I've seen at least 12 to 15 different indices for nitrogen use efficiency, which are all targeted for very specific research questions. But at the start of this work, 8 or so years ago, we didn't know what we should be targeting. So we've narrowed down, and we're defining in terms of what we're looking at for nitrogen use efficiency is nitrogen uptake efficiency. So this can be defined as the nitrogen acquisition from the soil by the roots. So the plant's ability to pull nitrogen from the soil, and in combination with nitrogen utilization efficiency, which is the amount of the nitrogen in the seed relative to the total plant nitrogen uptake. So these are physiological-based nitrogen measurements. There's also agronomic-based measurements, such as nitrogen use efficiency yield-based, which is based on the amount of seed harvested per unit of nitrogen input. So in order to take a look at, well, where do we focus? My colleague Raju Sullikanahali, which I saw in the virtual audience, I'm glad he could join us, came up with this concept of looking at a historical series. So what this historical series was a series of about 35 different lines, starting with Argentine, the very first Brassica napus rapeseed grown in the prairies, all the way up to modern hybrids. So hybrids that were released around the time that we had started this work. And this is just yield data, Statistics Canada yield data showing the yield response. So in collaboration with Rosalind Buchert from the University of Saskatchewan, we had a PhD student, Rajiv, who looked at this historical series over 2 seasons in Saskatoon and Melfort, and looked at them under contrasting fertilization, so low rates, or essentially what was available in the soil versus a high application of nitrogen, and looked at a range of different traits, including yield and nitrogen use efficiency indices. But what was really unique about this work was it involved a lot of sampling throughout the growing season to look at plant biomass and nitrogen uptake, as well as the partitions between the plant parts. And so when we dug into this data set, Rajiv had used a method called segmented regression to look at the patterns of yield and biomass, but also nitrogen use efficiency over time. So again, this is similar to the last chart, where along the bottom we have the years of release of different varieties, and then the responses, and similar. What we found was similar to yield-based nitrogen use efficiency, as well as nitrogen uptake efficiency, followed a similar pattern where there was an increase over time, but really a marked increase just before the start of the release of hybrids. What we weren't expecting was biomass followed a very different pattern in that biomass, this is largely the OPs, decreased over time, and then similarly, early hybrids had higher biomass compared to the more modern hybrids. And plant nitrogen as well as nitrogen uptake efficiency followed the same pattern. So our conclusion out of this was that there really hadn't been an increase in nitrogen uptake efficiency over the past 70 years of canola production in the prairies, although there had been nitrogen utilization efficiency increase. So that led us to the question in the current project of how can nitrogen uptake efficiency be improved? And Brendan did a nice job of introducing the germplasm and genomic platform that we're using to take a look at this. But in order to improve the trait, we need diversity for it. And that's where the diversity that's offered through the nested association mapping population is an exciting opportunity. So we've looked at the NAM founder lines. This is the fourth year that we're looking at them under, similar to what we did with the historical series, under low and high nitrogen conditions. This year, we're expanding out a little bit and looking at them under irrigation outlook. And similar measurements to what we did with the historical series. So looking at nitrogen uptake efficiency within segregating, mapping, and breeding populations is a little bit of a daunting process. And part of and the reason for that is because a lot of it's plant biomass and plant nitrogen based. And so we with sufficient resources, we were able to do a lot of physical sampling on the founder lines. But as we go into the real families, we go from looking at tens of lines to hundreds of lines, which means hundreds of plots to thousands of plots. And so we were happy to partner with the Plant Phenotyping and Imaging Research Center in the last round of funding. And Sarah Van Steenbergen, as part of her master's, she looked at developing NIR calibrations for tissue N, which saves us a lot of money just in terms of having a more efficient way to look at tissue N compared to some of the methods like LECO. And then another really exciting tool that she developed was prediction equations for plant nitrogen using multispectral data collected from a UAV. So this is just showing a prediction equation for total plant nitrogen. And with an R-squared of 0.83, that's really exciting, and that'll allow us to move into looking at this at a much bigger scale. So what is the diversity for nitrogen uptake efficiency across the founder lines? We're happy to see, again, a really good spread. Circled down here is NAM 0. So it's on one extreme in terms of being lower for nitrogen use efficiency or nitrogen uptake efficiency. Yet there's lines that are showing much greater amounts of nitrogen uptake efficiency. So similar to what Brennan explained, this bodes really well because we can go into the families generated from crosses with these founders to study the genetics and of nitrogen uptake efficiency. And we've also been looking at the data in terms of what lines are responding to nitrogen. So this is just what this graph is demonstrating here is the slope of the values just for nitrogen uptake efficiency, but it very much mirrors biomass, where except it's opposite for the values at low nitrogen compared to high nitrogen. And the reference line is this dotted red line at the bottom here. And what these other founders are, the ones where the slopes are significantly different from the reference line. So these are ones that are responding differently than NAM 0. And just wanted to dig into a little bit of data that we've produced over the past year. We have a subset of the founders which showed contrasting nitrogen uptake efficiency and nitrogen utilization efficiency patterns. Because even looking at, say, 50 lines under 2 nitrogen treatments, that's really a bit of an insurmountable undertaking in terms of, work, starting to look beyond just our main center in terms of different sites. So what we wanted to do was narrow down the number of founders that we're looking at to get a better handle on what the G by E is for these contrasting phenotypes. And this will help us in terms of understanding how to potentially use some of these traits deployed across a wider range of environments. So under Outlook last year, we looked at these lines under irrigation, and not surprisingly, the irrigation treatment really allows us to differentiate the different nitrogen treatments a little better. So in terms of outcomes, I guess the question as to why nitrogen uptake efficiency, well, hopefully I've demonstrated that there appears to be little or no improvement over the past 70 years of growing rapeseed or canola in the prairies. So how do we improve nitrogen use efficiency? Well, first we need to access genetic diversity. A major focus in what I presented today is accessing natural genetic diversity, and we also need new tools and methods. There's a reason why this approach hasn't been taken on segregating populations, at least in spring canola, and so we're able to capitalize on some of the really new and emerging tools, especially around digital phenotyping, in order to take this on in a way that we might not have been able to before. So what have we found? We found a wide range of nitrogen uptake efficiency and nitrogen responsiveness across diverse spring brassica and apis that do seem to be available for trait development. And what is the potential? So the germplasm, the lines we're identifying, as we dig into the rills, I think that germplasm will be of great value for the commercial breeding programs, but also the tools that we're developing. We're hoping that some of these tools would be feasible within commercial programs. And on the research side, some of the potential outcome out of this is opening up the opportunities for multidisciplinary study of genotype by environment by management. So, our collaboration with Melissa and Jennifer and Bobby is an example of that, where we can start to use genotypes to ask really specific questions. I think there's a lot more opportunity to push in that direction. So with that, of course, we are really thankful for the Sustainable Canadian Agricultural Partnership and the Canola Cluster and all the grower partners that are involved in funding this work and funding it in a sustained way with the second project. I do also want to call out to University of Saskatchewan collaborators who have been instrumental over the past 8 to 10 years just in getting to where we are today, as well as a lot of groups across all of Ag Canada and the Saskatoon Research and Development Centre and the associated farms. And this is just listing, I don't even know if this is a comprehensive list, but a lot of people who have contributed to various aspects of this project, whether it be the NAM, the field trials, the phenotyping.
Thanks everybody for joining us this morning. It's a real pleasure to be the, I guess I'll say the odd man out here a little bit, but it's as I said, a pleasure to be able to talk to you a little bit about the winter canola research that we've been doing here in Ontario for the last, almost 10 years now. So my name's Eric Page. I am a research scientist with Agriculture and Agri-Food Canada, and I'm based in Harrow, Ontario, which is the furthest south you can get and still be in Canada. And so I just want to give you a little bit of background knowledge. For some, it may be a new realization that we do, in fact, produce winter canola in Canada. I'm sure many here in the audience will be aware that the winter canola biotype, as opposed to the spring that's dominant in Western Canada, is the winter canola, by contrast, is the dominant form grown in Europe. And winter canola actually has a really good fit in Ontario. It's been tried several times in the past, so I would be remiss not to acknowledge that throughout the 80s and the 90s, there have been sort of a start-stop events with winter canola. And so we started looking at winter canola here in Harrow again in 2016 with some of the changes in germplasm And since then, we've had really good traction. And so we've seen a shift in southwestern Ontario from one producer who was interested in it and talked to us about it in 2016 to now having about 20,000 acres in Ontario, which is small relative to what we see obviously in Western Canada, and also even small compared to corn and soybean production in Ontario. But it's still a very significant increase. And why I would note it, and to put it in the context of Ontario cropping systems, for some time, Ontario has really been struggling and looking for that fourth crop in the rotation. And I guess I'm a firm believer that with the momentum that we have currently, winter canola can be that viable, profitable crop in Ontario to provide that fourth crop in that corn, soybean, winter wheat rotation. It fits really nicely. It complements well with winter wheat. Another point to note just on that 20,000 acres, we are currently seed limited. And I'll talk a little bit more about what that means in the next slide, but it's not for lack of demand. We sell out of seed every single year. It's just that we are taking a cautious approach in terms of how this industry expands throughout the province. So the map that you see here, I'm down there in the star, way down at the end, the north shore of Lake Erie. Detroit is north of me, so put that in context. This is the old winter kill map. It is being evaluated to expand winter kill coverage over into eastern Ontario, sort of south of Ottawa, the white spaces that you see up to the right. But just gives you a sense of where we can produce winter canola in Ontario. We have a really good, suitable growing region that stretches everywhere from me all the way up to Barrie and then sort of across towards Ottawa. And we have active producers growing winter canola in that region right now. So as I mentioned, I've been working with winter canola since really 2016. I should be mentioning here our seed partners. So C&M Seeds is here in Ontario, is the retailer. And Rubisco Seeds is a seed dealer out of Kentucky that we have a really good relationship with, Brian Kalbeck. And so they were both instrumental in registering the one winter canola variety that we have in Canada. Yes, I said one. It's a problem, but Mercedes was registered, full registration in 2020. So again, I talked a little bit about being seed limited. That's one of the reasons, is that we need more varieties registered. So we've been working away at developing a platform. So when I talk about a platform, I talked about, developing the agronomic information to help new producers be successful in growing winter canola. So we've done research on optimal planting dates. We've done research on tillage and residue management. And, where we're going with our support from the Ontario canola growers and the canola cluster is really talking about winter canola response to nitrogen. So where does a new winter canola producer go to get production information? They normally go here to the agronomy guide that's produced by OMAFRA. Again, I think this is an old photo. They've just recently updated this production guide. But the information that we have in there, and I work closely with Megan Moran, who is my colleague as the canola specialist in Ontario, to help develop some of the information that would go into this guide. What we have currently listed in there is recommendations in the fall for 40 pounds of actual N to help the crop establish. And then we come back in the spring with about 150 pounds to the acre. That information predates both Megan and I. We would love to know where that information came from and where the data was that was used to support it. But the goal of this project and what I'm looking at is really to produce new information. There's obviously been development in hybrids since then, and it's really nice to have Sally present what she did just prior to that, and Raju's work, how we've had change in germplasm and potentially change in nitrogen use efficiency. So we're looking to update this information with the germplasm that's currently being used. Okay. So overall, for this project that I'm talking about here, we're looking to determine a nitrogen rate response with Mercedes in particular, looking at biological and economic optima for nitrogen. We're looking at talking about where the nitrogen goes, so assessing nitrogen fate within the plant, so roots, shoots, seeds, etc. And then also looking at what nitrogen is left residual in the soil post-harvest. And so we hope to be able to feed that information. So Objective 2 really supports Objective 3, which is feeding that into some of the goals, climate smart goals that the government has been very actively promoting, looking at greenhouse gas modeling. And I'll talk a little bit at the end of my presentation about that. So to dive right in, I'm not much of a materials and methods slides person. I think I'd rather show you the data and explain to you what we did and how we got there. If you're looking at the figure on the right, so on the y-axis is spring applied actual N, and I apologize, I've switched to metric here. It's in my head, that's what I think, but I'll try to reference the pounds to the acre if that's where your head's at. On the y-axis is canola yields in kilograms to the hectare. Just a little point out here. So the yellow arrow highlights what our current recommendation rate would be on this nitrogen response curve. And so that's 168 kilograms to the hectare is about 150 pounds to the acre. Okay, so that's our current recommended rate. All of these treatments had fall applied at 40 pounds to the hectare, so they're all even in the fall. This is just based on spring applied nitrogen. So the structure, that's really the structure of the experiment that we're running here. We're doing it at a couple sites here in southwestern Ontario. So I mentioned that I'm based in Harrow. Harrow is a sandy loam, so it's a really nice soil type to work with. A little north of me is our satellite site. It's called Woodsley. And at Woodsley is representative of sort of the north half of southwestern Ontario. It's a heavier soil. It's a Brookston clay. So there should be contrasting responses here, and that's why we have it at the two sites. I'm showing you Harrow data here. So this to the right is Harrow data. So again, we're looking at quantifying productivity, but also nitrogen use. When you start talking about yields, I will say these are small plot yields. So just for reference, they're at that current recommended rate. So that fourth point from the left, that yield is about 5,000 kilograms to the hectare, which is about 90 bushels to the acre. It is small plot, but I will put for context out there that most of our experienced producers, so guys that have been growing winter canola for 3 to 5 years now, their field averages probably are north of 80 bushels to the acre. And one of the reasons that canola is gaining traction is really those nice high yields, and many of them will follow that yield of canola with double crop soybeans, and they'll go 40 bushel beans after, which is a very profitable acre. So in terms of discussing the data that I'm showing to you here, so the graph is still the same. I just wanted to walk you through a couple of points that we're going to be looking at through this type of yield response curve. So it's a quadratic yield response curve, very standard. This has been done with a lot of other crops. It's not new. This is just simply a reapplication of the data. It's important to highlight that what we want to do in our structuring of our rates here is to make sure we have enough spread to be able to get an optimum point. Where does that curve, the benefit of nitrogen sort of top out and then start declining? And in this particular site year, so one site year data, the biological optimum is about 304 kilograms to the hectare. So that's a heck of a lot of nitrogen. That's where you have your maximum yield in this environment, this particular environment. I wanted to highlight that because where we're driving is looking at a different concept. It's called the maximum economic rate of nitrogen. Again, it's not a new concept. It's well established in the literature. Dr. Bill Dean at Guelph has used it on several crops. But really what it does is a very simple economic analysis of looking at crop value versus cost of N. These are the numbers that I used in this particular calculation. They were representative for the 2024 growing season. And this is where the MERN sits. MERN is maximum economic rate of nitrogen. So where is the most profitable rate of nitrogen for a producer, just based on those two simple things? Now, I know Sally had a great graph there of showing the various, input costs. Nitrogen is, if I remember correctly, around 15% of the input cost. So there are other things going on, but certainly this is a starting point for producers to understand, where is that best profitability lining up. And in this particular case, it's about 211 kilograms to the hectare, reminding you again that the dot directly to the left at 168 was our recommended rate. So not far off. A little bit higher would have been better, but pretty close, okay? So the current recommended rate turns out to be about 97% of the murrin. So I think that's a pretty acceptable mark. And also just to highlight that your rate of nitrogen return as you move there from left to right on the graph really declines, right? More nitrogen when you get beyond that point, you start hitting the top of that quadratic curve it's incrementally less beneficial, okay? So part of this project is not just looking at the yield response. Obviously, that's a very, very, very important part. But we also want to take a little more holistic look. We know that there is trade-offs with higher nitrogen. And again, this is not new to winter canola. This is more generating data that I think we already have for spring canola. But we have an opportunity with that end rate structure. So again, it's the same graph mostly. The x-axis is still the same, still spring applied nitrogen, same rates. Y-axis is oil content in this particular case. So it's just an opportunity to demonstrate that really what we already know, or to reaffirm that, higher N rates do have an impact on our oil content. And in this particular site year, it's a really nice linear relationship. Oil content declines by about 1% for every 100 kilos of nitrogen applied. What we do with that information is still up in the air, but I think it's important to have, again, a holistic picture of when we're applying more nitrogen, what are the consequences for that action? As I touched on earlier, we're obviously still looking at nitrogen use and nitrogen fate, both in the tissue and then residual soil and after harvest. And so I'm going to move to some of that data now. So what's left after harvest? Very big question for us. And I think this particular structure of experiment allows us to sort of look at that very nicely. Similar graph, again, x-axis is still the same, spring nitrogen applied, except this time on the y-axis, this is plant available nitrogen in the soil after harvest. We did do a fall baseline soil sampling, so that's what you're seeing there in the gold line. And then everything else is what is remaining after harvest. I think we'd all agree that a less leaky system with nitrogen is what we all strive for. So ideally, N in equals N out. But what this graph is really driving home for you, and I will say this has always been Harrow data that I'm showing you. It's the same site year in all the figures. In reality, more N applied, there is more N remaining. So when you get out beyond that recommended treatment, which was at 168, that's our current recommendation, so the fourth dot from the left, you really see it's an exponential curve. Exponentially more nitrogen is remaining as you apply more nitrogen beyond that point. So again, part of changing our thinking, or maybe just realizing the consequences if we are going to apply more nitrogen, what are some of the consequences? And again, this is all really to feed into information into greenhouse gas modeling, or sorry, that's one of the end uses. It's to communicate that information in terms of a farmer producer making decisions about nitrogen, but it's also to help parameterize the model. So winter canola is maybe, certainly in Canada, we do not have the baseline information to parameterize a model like HOLOS. So Dr. Roland Krobel with AAFC is leading that model development. And so in consultation with him, we're going to try to make sure that we have the data that he needs so that he can incorporate winter canola as an option into the HOLOS model, and so that we can really see some of the model projected impacts on things like greenhouse gas emissions from changes in nitrogen practices based on the data that we're generating. So that involves residual N after harvest, which I just showed you, but it also involves tissue N content, so in roots and shoots, and that's something that we've actively been doing very recently. So just for everybody's sake, and we understand our production cycles, so currently where we're sitting right now is we will be making a decision on desiccation for winter canola probably by the end of the week, desiccating maybe Friday or Monday, and our combines are going to be rolling probably about 4 to 7 days thereafter. We'll finish winter canola harvest in early July, and then we'll be actively looking at planting our subsequent crops, and that's what most producers in terms of their production cycles are going to be doing right now. So it's about to get real busy for us real soon. So I was really happy that the timing of the meeting fell when it did. So just in summary, what does this really mean to a farmer? To a winter canola farmer, I do get a lot of questions about, is my N rate right? To now, I haven't had any data to be able to answer them. We can look at the European data. We can say that this is what the winter canola N rate response is. We can look at the quality data from spring canola, but we haven't had sort of Canadian data on winter canola production yet. What I would say with 1 year of data, the second year about to be in the combine very soon, is that our current recommendations, at least in Harrow, aren't bad. 97% of the MERN, which was the maximum economic rate, is if a farmer had applied the current recommended rate, it was pretty good in that site year. I'm very interested to see how that holds up over the years of this study, and also how that holds up as we change soil type, because we very much know that N rate response is going to be impacted by soil type. I think it goes without saying that, more nitrogen means higher yields. But there are trade-offs, and one of the trade-offs that we're just highlighting here is already well established, but we're reaffirming that, is that you do decrease oil content fairly significantly as you are increasing nitrogen content. So just something really, I think, the industry to consider. Messaging at the end to farmers: optimize, don't over-apply. I think we can clearly demonstrate that, reducing inputs results in less residual soil N. If that's our goal, and we're trying, to reduce either leaching or volatilization, I think that should be a real take home. And hopefully with the data platform that we're generating here, we can provide improved recommendations for farmers where they can see that trade-off between, the economics as well as the environmental impact of their fertility practices. So with that, I want to thank certainly everybody here in the audience for listening, and then the SCAP for funding this project. Ontario canola growers have been fabulous to me and supported me all the way along through this project. Really excited to have their support in developing this industry, and also the Canola Council of Canada. We had the pleasure just very recently of having a visit from Dr. Gilles Sandon, who is our retiring ADM. It was a chilly day, that is May 8th. Canola was in full flower. I don't often get people that show up at the right time. My tours are always off sync with everybody else here. So it was just a real pleasure to have him and Elena Yule visit us and see the results of this project. So with that, thank you, and I'll turn it back over to Chris.
Okay, great. Thanks, Chris. Thanks for the invitation to speak here today. I'm gonna present our canola cluster project, Precision for our management, improving nitrogen use efficiency, greenhouse gas emissions, and production economics of canola. We have a large team here. Myself as the lead, Philip Harder with Croptomistic, Rich Farrow from the University of Saskatchewan, Chris Hozapfel from Indian Head Agricultural Research Foundation, and Tristan Skolrud, our economist at the University of Saskatchewan. Firstly, I'm coming from the University of Manitoba. Not quite its campus. I'm at home right now, but nevertheless, this is still very relevant. Are located on original lands of the Anishinaabeg, 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 Dakota Oyate, and Dene, and on the national homeland of the Red River Métis. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in the spirit of reconciliation and collaboration. Now, some background here. Growing interest in 4Rs of nitrogen management, the right rate, source, timing, placement and fertilizers to increase nitrogen use efficiencies. We've been studying this for quite a couple decades now in various crops here in Manitoba, and it's really picked up 4Rs applications right across Western Canada and Central Canada as well. We have a great deal of understanding of major 4R practices on N2O emissions and field crop yields. And N2O emissions, of course, being a greenhouse gas as well. However, in Western Canada, many fields are variable in soils and topography. They have undulating landscapes, and that affects the return on investment of nitrogen fertilizers and changes nitrogen losses response to fertilizer application, depending where you are in the landscape. Nitrous oxide emission is nitrous oxide is emitted more so in depression areas because it's wetter, and so we know that certain areas of the landscape have greater potential for N2O release than others. So when we're managing a strategy to reduce emissions and where some 4R practices cost money and potentially even actually steal some profits, it's important to know, could we tailor the 4R practices to those regions or areas of the field which gives us the most return in terms of greenhouse gas reductions and also less impact on profitability or reducing. We do know that nitrification inhibitors greatly reduce N2O emissions, anywhere between 30 and 60%. It's pretty dramatic. We do know that. And here, with this cluster project, it's the first examination of what I've coined the word precision 4R practices in heterogeneous landscapes. So what we're doing here is applying variable rate nitrogen application and variable source of nitrogen to reduce nitrous oxide emissions and improve or maintain yields. And one of the key technologies that we're using here is actually a Canadian-based technology, SWOT maps. This is where Croptomistics and Philip and others really come to play as major, I don't call them industry partners, they're collaborators. They're right in here in this project. Now, SWOT maps can provide precision prescriptions of fertilizer additions based on management zones in a field. And a field is broken up or segmented into 10 different management zones. You can see an example here. It's based on, for visual representation, based on colors. Red really means the highest portions of the field, so the knolls. And green, deep green, are basically depression areas or lower topographic units. Now, those 10 management zones, and every field would be broken up into 10 management zones. This is the way how the process works, is based on the soil properties throughout that field, the water regime, in terms of how water moves, drainage, stuff like that, and the topography. So at heart, there's an electromagnetic survey, and the image on the left is the SWOT box, EM survey, which really looks at, tells you about the texture of soil, and in particular texture, can also salinity, and a digital elevation model to get the topography. And with that, there's ground truthing to sample areas, looking to confirm that there's differences in soils. Take some samples, lots of samples actually, for nutrient analysis, find out what's present in the soil before planting, and doing a yield analysis in terms of seeing what has been the previous responses to nitrogen fertilizer in these different management zones. And they've used all that to fine tune a prescription map. Okay, for, in this case, what we're focusing on is nitrogen prescription map for these different zones. And more on this is where the precision comes in. It's really an extension of variable rate nitrogen prescription mapping to include variable source of fertilizer. For this project here, it's really mixing up urea, granular urea, conventional, and super U, which is a nitrification inhibited granular urea product. And what it is basically is, in those high areas, those knolls and crests, they receive urea. In the depression areas where there's more prone to be lost, such as through denitrification, leaching, nitrous oxide, so forth, that is where one invests in the higher cost Super U. And you can see here, that's where the Super U goes. And this is an example from 2023 for our pre-cluster shot at research so that we can get it really good in 2024. You can see in this case those depression areas calling for very low nitrogen, and it's because the yield potential is hampered because of the moisture. So there's that variable nitrogen in there, and this is amount of product, not amount of nitrogen. And so you can see in management zones that are intermediate between 4 and 6, there's a blend. There's a blend of those two nitrogen sources. We have three objectives for this study. Understand if variable rate N applied based on management zones improves nitrogen fertilizer use efficiency of canola. If N2O emissions are reduced by inclusion of a nitrification inhibitor in high emission management zones, basically in those lower landscape elements. And what is the economic benefits of precision for our approach? So in fact, we want to know, hey, not using Super U and spending the extra money on Super U for the whole field and just in the areas most prone to nitrogen loss, is that financially beneficial to the farmer? In other words, it would reduce their input costs by not using a product like Super U where it's not needed, like in the dry areas on knolls. Okay, it's a 3-year collaborative study. It started last year between 3 research institutions, U of S, IHARF, and University of Manitoba. Prescription fertilizer service provider, and that's Croptomistics, using their SWOT maps approach. And all 3 canola grower associations of the prairies under the umbrella of the Canola Council. And the research is done on farm fields with commercial fields. And the research is actually, in terms of the agronomics, treatment, application, and so forth, is actually done by farmers. And those 3 sites are near Saskatoon, Indian Head, and Brandon. And just to give you a bit of an example, not an example, but these are the actual treatments that we're using, 5 treatments. And what we do is we apply the treatments in strips across the field. This is an example from 2023 near Brandon. And the different color code of the strips matches up with one of these treatments up here. And we have the typical farmer practice, which is just a flat rate of urea based on general soil test. Second treatment, variable rate nitrogen. So this is VR nitrogen application, whereas urea nitrogen rate is based on SWOT map prescription. And then we get into the precision, which is variable rate urea and Super U mixture, and the changing mixture, depending on zones. And then treatment 4, the same as the precision or treatment 3, but 10% less nitrogen added, total nitrogen added. And why 10%? Well, it's because that's basically what the cost of these enhanced efficiency nitrogen fertilizers are. They cost more, and they're supposed to reduce nitrogen losses, and they do. And so the theory is, and well, it's not a theory, it's a hypothesis, that's saying that, well, if we're saving nitrogen and not losing it, then we don't have to apply as much nitrogen, because farmers apply nitrogen at a rate that accounts for nitrogen losses. And the 5th treatment is a control of no urea or Super U. There is nitrogen in terms of a MAP, as the farmers apply that to their whole field. And what you're seeing here are actually 3 elements. The knolls in red across this field, the intermediate or middle landscape positions in this light green, and and the dark greens, the depression areas. And what these stars represent are actually locations that we sample for gases, for nitrous oxide using static vented chambers manually. So our poor students have to run around the field. And so they get a lot of steps in the course of a day. And then when the canola is higher or taller, like in July, then it gets really tough to walk and stuff like that. So it's a tough job. It's a lot of ground to cover in this experiment. A lot of sampling under really tough conditions. And here's an example of nitrous oxide emissions in our spin-up year in 2023 that we only did at the University of Manitoba. And this is by Brandon on a commercial field. And we have the depression area in this column, the middle landscape unit, and the knoll top. And these are the N2O emissions over time. And these are the treatments here, the flat rate urea going down to the 10% less precision end. Now this year that we was forgotten to put the controls into the GPS computer on the cab, and so we didn't have a control in 2023. But one thing that you can see is that depression areas had much more N2O emissions. Early on, it was a very wet spring with high snow melt and a high water table in this field. And you can see some reduction in terms of going VR, urea, and definitely down here with the Super U and less nitrogen. But however, once the soil started to dry up, we still got the nitrogen being released. I think outside the N2O being released. I think what happened here is the inhibitor wore off because it does degrade. And it was wet, so it didn't nitrify. And then when it started to dry down and the nitrifiers got in there, the inhibitor was dissipated and gone. And hence we got the N2O. So that's my running thinking on that. But you can see the other landscape positions, much lower N2O, and some effects in terms of the treatments here. And in these numbers here are the cumulative N2O emissions that were released over the growing season. And let's go to Indian Head last year. Very clear, interesting results here. This is the depression area, lots of N2O emissions. As soon as we go to VR, boom, goes down. And then when we do the other treatments, goes even further down. So that's pretty spectacular results here. Quite a drop there in terms of N2O emissions. It's like 80% reduction. The middle zones here, we see the benefit of the VR and precision. The precision and the precision reduced rate. At the knolls, not as clear, especially because the VR rate of urea went up for some reason. So interesting. Now we look at yields here at that Indian Head. Here's the controls, and first these are the different management zones, 10 of them. You can see there was a nitrogen response. Here at the crests or the knolls, there was a declining yield with the practices. So this was the flat rate here, and this is more and more precision 4R and lower N rate. Somewhat pattern in the second. So that's kind of not quite the shoulder, but getting there of the slope. And then we start getting to the middle landscape positions, kind of flattens out a bit. And then we see this pattern again and down in the depressions of a lower yield. So it's very interesting overall for the field when it's weighted. You can see the flat and the VR nitrogen are equal, and those precisions are lower. So in terms of yield, I could have hoped for something different. In terms of N2O emissions in Saskatoon last year, you can see reduction in N2O emissions with the precision practices. Again, here in the mid slope, that VR urea, in this case with the VR, there was actually adding more nitrogen because the yield potential in that landscape element is really good. So that's what's happening there. So it's interesting in that when we see this reduction here with the precision practices, that's quite a reduction actually in terms of N2O emissions with the precision. And we look at the yield in Saskatoon, some slightly different patterns. As you can see here, increasing yields with the precision practices in zone 3. So that's around the shoulder position of the knolls. Same with 4 and 5. And then we get this pattern of reversing. So we get to close to the depressions and lots of variability in the depression itself. So as you can see, in terms of yield response, it's going to be different for the elements. And really what's going to be key here is the spatial amounts. Let me throw back here. You might have saw this because these elements have different proportions in a field. So it's really the ones that have the most acreages that may have the most overall impact on results. The Brandon yields, we see this pattern again going and declining at around the knolls, and but in the depressions the other way around, with a precision 4R being beneficial. Okay, so it's only the first year. So, but we have 3 sites. We have 2 more years, another 6 site years. So looking forward to it. Now, if you got some time on your hands, July 30th. I'd like to hear more about this, actually see in person the site. We're having the Manitoba Precision 4R Field Day. The first time we've done this by the farm field, just northeast of Forest, Manitoba, just north of Brandon. So we've got a good roster here on the program, including the producer that is cooperating with us. And so if you want more information on that, please let me know. If you want this flyer, I'll send it out to you. Now, in terms of acknowledgements, lots of people to thank. Very expensive work, very laborious work. As you can see, 3 research sites. We're dealing with whole fields here, lots of players. So thanks to AAFC and AgriScience program, Sustainable Canadian Agricultural Partnership, for that government funding, for the matching funding, Alberta Canola, Saskanola, and the Manitoba canola growers. And then in terms of plethora of individuals, the University of Manitoba, Brad and my crew at IHARF, Chaturi and Danny, at the University of Saskatchewan, Frank and his team there, and Croptomistics, Corey Wellness, who's the president of Croptomistics, great supporter of this project, and Amber Nags, who's a local Croptomistics rep in the Brandon area. Koch Agronomic provided the Super U for the 3 sites, and of course the 3 farmer cooperators for their land and for doing the actual operations. So with that, thanks, Chris.
Yes, so thank you, Chris, and thank you to Brendan, Sally, Eric, and Mario for those excellent presentations in our first session. Really set the bar high for the day here. I'd like to remind everyone, if you do have questions, to please type them into the Q&A chat there, and we'll do our best to get them answered for you. Thanks to Jay Wetter here, who does have a few questions in the comments, so we'll maybe get right to it. So the first question was for Brendan. Just wondering if you could conclude, based on your NAM results, that the seed produced in Chile could have low cool season vigor when planted in Canada the following season. Any thoughts there?
Yeah, as I respond in the chat, we're not drawn to that conclusion right now. It's definitely something that we're interested in, but based on the entirety of the data sets we have, which go beyond what was shown today, that trend doesn't pan out across all the data sets. So we'll continue to look at it, but the answers can be more complicated, involve more factors, we believe, than just the Chile growth location.
Excellent, thank you. Sally, I do have a question for you. The NAM population has been brought up a couple of times. For those who haven't had the opportunity to see that in the field with you, could you just give a little more detail on that population and how it does encompass the genetic diversity that's useful in this research?
Yeah. So the population is comprised of 51 parental lines. So our reference line NAM0, it's a line that came out of my predecessor Gerhardt's breeding program. So it would, if there was a market for OPs into the 2000s, it would have been released alongside, say, Q2 and some of those late OP varieties. And it's an early flowering, really yield stable variety. So that was chosen because we knew what it was going to be crossed to was a lot of really diverse material. So the 50 founder lines, this represents the range of diversity we can find all across spring Brassica napus. So not the biennial type that Eric was talking about, but the spring habit. So for this, Isabel, Steve, and I, we looked into all kinds of germplasm collections. So we looked through what was available through Plant Gene Resources Canada for Brassica napus. We looked through other diversity collections that had been put together in other parts of the world. We looked through collections that we had at the Saskatoon Research and Development Center. And so using a combination of techniques, looking at the genetic diversity, like based on, it was a combination of SSR markers and then some SNP markers at the time. And the phenotypic diversity that we'd seen in field trials or that had been characterized by other people. Some of the lines were maybe had some interesting traits about them, but they were representative of, say, so for certain lines from a different country. So the origin lines, I think about a third of them are Canadian or European, and then the remainder of the 50, a lot of them come from Asian type origins. So it encompasses a lot of genetic diversity that may not be present within breeding programs or may not have been selected for within modern breeding programs. Typically, when you have crops that have very strict quality, seed quality parameters like canola, you tend to select really hard for what you need in order to register things, and then you can lose some genetic diversity along the way. So the goal was to open it back up and see what else is there that could be incorporated in with the appropriate tools.
Yeah, excellent. It is for anybody who does have the opportunity to see it in the field. It is well worth seeing. It's pretty impressive. So let's move on here. Eric, Jay had a question for you here. Rather than thinking about oil percentage per kilogram of nitrogen applied, would the industry be better suited to approach this as looking at oil tonnage per acre for the crop? And really that comes to rise because farmers don't get paid on oil content.
Yeah. And as I said in the chat there, Jay's absolutely right. I had a slide in there previously. I trimmed it basically on this concept, and it really could be a win-win if there was an oil pricing scheme. Like if the industry, if the processors wanted to optimize oil production per acre, I mean, one way to do that would be an oil pricing scheme, because as you know, farmers are paid on bushels, they're not paid on oil. So to drive them to optimizing that oil quantity per acre, you probably would have to incentivize it. I very crudely did that, and what I can say is that, from what I showed today, you obviously change your economic, your MERN, your maximum economic rate of return. You're actually decreasing it, there's a lot of assumptions there based on the pricing scheme that you're using, but in theory, you could do it. So both achieving an optimum nitrogen or an oil tonnage per acre for the processor, and also choosing a maximum economic rate that results in the highest profitability for a farmer. Those two things aren't mutually exclusive. They could actually work together if that type of scheme was implemented. And it could potentially have the benefit of reducing N rates in terms of environmental impact as well.
Thanks, Eric. So, kind of on that topic of the maximum economic rate of nitrogen and looking at it from a 4R standpoint, I was just
Yep.
curious, what timing were you applying that spring application? And I
Yeah.
guess subsequent to that, or additionally, if that timing was changed, do you think there would be any changes to that maximum economic rate?
Yeah, excellent question. And the subsequent question should be, did I apply an inhibitor at the same time? Um, yeah, no, I was thinking that with when Mario was presenting. So yeah, with winter canola being off cycle to spring, and so when we are applying our nitrogen, as soon as we can get on the field. So I'm pushing for late March, early April is normally my timing. It's pretty cold then. So to achieve this project, we did stream 28% to get our spring nitrogen rates. There was an inhibitor included at that point in time. In my very brief conversations with Dr. Craig Drury, who's our local end specialist here in Harrow, does a lot of the greenhouse gas modeling for corn production down here. Um, Unclear as to what kind of volatilization or end losses we might have given the temperatures at that point in time in the year. Could it impact the MERN? Absolutely. If we are in fact losing nitrogen at that point in time, I wonder if we might be losing it more to leaching with spring rains than necessarily to volatilization. But I'm not an end specialist. I will fully admit that and defer, even if Mario might have a comment to follow up on that.
Any thoughts there, Mario, before I get to your next question I have written down here?
Well, yeah. Well, with the application with a crop already growing, usually it's a top dressing or a dribble application, something like that. So the most efficient means of getting the nitrogen to the plant would be in the soil. And that's a little bit more challenging when you have a plant already growing there. So we didn't do it with winter canola, but we've done it with spring canola, if you want to call it spring canola here, where we've done split applications where we've dribbled UAN on at the rosette stage. And we found good responses to that, basically similar responses in terms of yield as application in the spring. It just gives you a bit of insurance to not lose as much if you had lots of rains and before the canola was really taking things up. So, I think it's something like basically like a new crop for us, which would be winter canola that Eric is working with. These things would all need to be worked out, right?
Yeah.
And inhibitors can be, in terms of in this case, a urease inhibitor would probably be the most helpful. In case there wasn't rains right after that top dressing or that dribbling that went on. Just give you about 3 or 5 days insurance that you want to get much volatilization until the rains wash the nitrogen into the ground. You could go with UAN, which has ammonium and nitrate present and a smaller portion of urea compared to granular urea, and that would reduce the volatilization as well. So yeah, there's opportunities there. Yeah.
Excellent. Thanks, Mario. So, Mario, with regards to your Precision 4R project, one of the challenges I always hear about for uptake on new practices is logistics and equipment, in addition to cost. So, just wondering, in your opinion, what's the feasibility of this Precision 4R approach at this point in time?
Yeah, it's a good question. That hurdle is becoming smaller and smaller as time goes on. So as producers are replacing their equipment, the new equipment has for obviously for precision placements in terms of GPS and they can do things, make several passes actually, and do things geospatially very exactly. In terms of variable rate, that's a no-brainer. That's easy. The equipment basically comes outfitted like that. And the VR and precision 4Rs, it's going to be those producers that are going for it. Those producers that are big acres, they're going for that economy of scale, and they're looking for edges. And frankly, they like technology as well to help them out. It's fun for them. So I don't see it. It's going to be those folks that are going to be the ones that this technology appeals to, and they'll have the equipment. Okay, the ones that don't have the equipment, that are dealing with seeders and drills and things like that from the 1990s and so forth like that, they're going to retire that way, and they're not going to be doing this. So anyway, I'm very optimistic. I see the young folks. They love this stuff, and they're there with their iPad. Everything goes into an iPad, and then the iPad gets plopped into connected in the cab and links up, and then it's easy peasy for them.
Yeah, so the opportunity is definitely there, and I think, like you say, uptake will continue to increase as more people adopt it. So excellent. I do see one more.
But Warren, sorry, it's going to be that return, right? That it's the return on investment. That's going to be the key thing, because it's fun for them to tap around on an iPad and do things like that. That's great, but they'll want to see some return for that. That'll be the key thing. That's kind of why we're doing this project, right?
Certainly, yes. Moving to that targeted approach, I see value there. We've got one more question. We'll try and get to it quickly here, and then turn things back over to Chris. So I think this would probably be going to Sally. So it seems odd that the NAM0 is characterized as a poor germinator. Wouldn't this trait have been selected out if it's a good variety, yield stability, early flowering, etc.? Does this poor germinator description indicate its average with 50% emergence under most conditions, or is it truly inferior for this trait? I guess Sally or Brendan, that could be directed to you.
I'll start, and then I'll pass to Sally. So in the lab, NAM0 is only a poor germinator under cold conditions. At temperatures warmer than 10 degrees, its germination is not remarkable. In fact, all the founder lines germinate 95% or so or greater at 20 degrees. But in terms of field emergence, maybe Sally can comment on whether there's any patterns emerging in that data with regards to NAM0.
Yeah, and overall in the field, it is a good emerger, which is why it wasn't selected against. I think what's interesting coming out of this work is that we're finding, especially several of the diverse lines, there's different thermal dependencies. So Rob Golden can speak to this best, but and this is, I think, coming from a lot of the wheat science research, where they look at the range at which optimal germination happens. And I think what we're seeing in terms of the data is that there's some evidence that across the diverse collection, there might be different thermal dependencies or ranges in which different genotypes will germinate. So yeah, but yeah, in the field, it germinates well, it emerges well. So.
Excellent. Thank you for the clarification there. And I know we started a few minutes early and we're ending a couple minutes over our time here, so I'll maybe turn things back over to Chris, but thanks again for those excellent presentations.
Thanks, Chris. So I'm going to talk about a project which has been incubating for quite a number of years, and I'll give you a little bit of background on that. The topic is really biotechnological. It's we're using gene editing to improve canola yield, but as a result of some of the work we've done, we've also enabled us to protect that yield and increase stress tolerance in the plants that we've produced. The work has been conducted with colleagues here at Guelph, Michael Eames and Li-Ping Wang. In particular, Li-Ping Wang has been involved in creation of the transgenic canola that I'm going to discuss and talk about in a little bit more detail. When you think of improving yield and an oilseed crop, the logical thing perhaps to consider would be to manipulate the oil biosynthetic pathway, but that's not what we've done. What we've done is improved the yield of this plant via an indirect mechanism manipulating starch biosynthesis in the vegetative parts of the plant, and this has been done via mechanisms involving alteration of sources and sinks by mechanisms which we're not yet still understanding. So to give some background, this cartoon illustrates the importance of starch in all plants that store starch, and that's most of them. And in particular, canola is a plant which has high levels of starch reserves in its leaves, and those starch reserves serve as temporary stores of carbon. Around 30% of the carbon that's fixed in photosynthesis is converted into starch as a temporary store, which is utilized during the nocturnal period for important processes. Respiration and growth and development take place during the dark hours as well. So starch is an important carbon sink, but it can be a source of carbon when it's being released for growth processes during all the way through the light and dark periods, but in particular during the dark period. And starch reserves are not just deposited in the leaves. This is a cartoon of an Arabidopsis plant, and I want to emphasize the very, very close relationship between the model plant that we started this work with, Arabidopsis thaliana, and canola. They're very, very closely related. And what I'm showing you here in this cartoon format also applies to the canola plant. So that starch reserves are not just deposited in the leaves, but also in strategic locations in floral tissues and saliques, and they act as I said, as a temporary store of carbon in these important sink tissues. And of course, the most important sink tissue in the canola plant is the oil seed itself. And you can see in this cartoon, and we can show, and I don't have time to show the actual stains of and the canola plant, but we have significant starch reserves being produced in and around the salique and in the developing seed as well. The background work I just want to very briefly mention was conducted in the very closely related plant Arabidopsis thaliana. This was some years ago, where we introduced a different type of branching enzyme, which is an important enzyme in the starch biosynthetic pathway. We started off by removing the endogenous branching enzymes. You can see on the in the left, in the wild type, which is stained with starch using iodine. You can see the black image on just to the right of that wild type. But if we remove the two branching enzymes that are present in Arabidopsis and replace them with branching enzymes from maize, we can complement starch biosynthesis. So removing the branching enzymes, SBE 2.1 and 2.2, creates a plant which is small and unable to produce starch. And that also underpins the importance of starch in plant health. But if we complement this mutant with branching enzymes from maize, we restore starch biosynthesis, and you can see on the right, we restore biosynthesis, but all starch biosynthesis in the darkly stained leaves, but you can also clearly see an increase in biomass. And if you look further to the right in the graphs, we show that those plants that have been complemented with the branching enzymes from maize have increased oilseed yield and increased salique numbers, significant increases in those numbers. The importance of the branching enzyme that we introduced into these plants was because the type of starch that's produced is a storage starch and turns over differently than the transient starch that is present in the leaves of the Arabidopsis and the canola plant. So this is the basis of the experimental work that we've done in canola. If you could move forward, please. And the strategy has been similar to what we did with Arabidopsis, which is a diploid and has 2 copies of the branching enzymes. Canola, an allotetraploid, has 2 genomes, therefore 2 copies of each of the 2 branching enzymes. So there are 4 branching enzymes to knock out. And this was conducted by Dr. Liping Wang using CRISPR-Cas9 mediated gene editing. And this involved loss and editing of each of the branching enzyme isoforms, creating a null mutant called a sextuple mutant, which forms the basis to produce the transgenic plants where we expressed the maize branching enzyme. And the data I'm going to show you is for expression of the branching enzyme 1 isoform from Zea mays. We've also conducted experiments, but I'm not going to show you, with the branching enzyme 2B isoform from Zea mays as well. And then we're in the process of characterizing these stable lines under controlled environmental conditions, and we're also conducting field trial evaluation, which I'm about to, which I'll show you something of in a moment. So forward again, please. So as I said, in theory, this involved the editing of 4 genes using this Yao promoter, and was complicated by the fact that there are gene duplication events in the canola variety DH12075, which is amenable to transformation, but nevertheless had two extra copies. So we ended up having to undergo 2 rounds of gene editing. Next slide, please. And you can see in this table that there's several rounds of gene editing were required to remove the branching enzymes from this plant. Next slide, please. The plants themselves can be seen here, and although the change in phenotype is not as striking as it was with Arabidopsis, you can see clear evidence of an marked increase in biomass in the transgenic plants. There are 2 wild type on the left and 2 transgenic plants on the right expressing branching enzyme 1. And we're also seeing in the sextuple mutant similar phenotypes as well. And I'm going to talk about those in a minute. The most striking part is an increase in biomass, a thickened stem, and increased branching of aerial portions of the plant. And you can see on the right, the 2 plants are not only bigger, but there are more branches holding, subtending flowers, increased flowers, and increased calyx. Next slide, please. The data from these, so we have some data from controlled environment conditions where the plants expressing branching enzyme 1, the transgenic canola, has been assessed for a number of agronomic traits, and you can see clear and marked differences in the numbers of stems. I'm working from the left to the right. Increased numbers of stems up to 60%, increased numbers of calyx up to 40%, and a total seed yield per plant increase of 35%. An important trait that we noticed in the sextuple mutant and has been continued in the transgenic plants is an increase in the cross-sectional area of the main stem. As you can see on the bottom left, a marked increase in stem diameter, increased above ground weight, and increased weight of each single calyx. So marked increases in yield resulting from the application and expression of the branching enzyme 1 isoform in canola, which is very interesting, very exciting. Move the next slide, please. So on the left, you can see this is the data for the sextuple mutant lacking the endogenous branching enzymes. I want to emphasize that this has been retained in the BE1 transgenics, but the data I'm just showing here is for the sextuple mutant. And you could see on the extreme left, the increase in stem diameter, and on the right on that section, the photograph, the actual cross section itself of the sextuple mutant is larger. So these plants have thicker stems and potentially increased phloem for movement and mobilization of carbon reserves around the plant, and also a potential for reduction in lodging as well. And that's something we're going to test, but we haven't tested yet. On the right, you can see data for on a preliminary experiment that has been conducted under controlled environment conditions, where we modified water status and heat. And you can see, and the data is for the sextuple mutant. We've yet to do it for the transgenics. So the wild type is in blue, the sextuple mutant is in white. And you can see under control conditions, there are similar total seed yield per plant is similar, not quite as high as the wild type, but this is the sextuple mutant we're talking about. But when these plants are droughted, you can see there's a increase over the wild type in seed yield per plant, and this is extended further under heat treatment. So the plants are able to withstand drought and heat better with this thicker stem. Next slide, please. Just to emphasize, and these are transgenics we're talking about now. These are canola plants with the expressing of the branching enzyme 1, the wild type plant on the left compared with 2 examples of the transgenic. And you can clearly see the thickened stem phenotype and increased floral branches. These are plants that have obviously been cut away, so you can see the stem difference. And this is partly a result of increased stem lignin and stem cellulose contents, as you can see on the data on the right. The next slide, please. An important thing I just want to mention is that, as with the Arabidopsis model system, in the transgenic plants, the transgenic canola, oil seed quality has not been affected at all. So that's an important thing to note. We've been manipulating source-sink relationships, and the effect on oil seed quality has been unaffected. The next slide, please. We're also very interested in trying to understand the biological mechanism that underpins these changes. And this simplistic slide is just to illustrate that we're undergoing some RNA-seq analysis of these plants. And we've shown that there are a number of upregulated and downregulated genes. One thing I just want to mention that's of interest, and we're going to pursue further, is that in the plants expressing the branching enzyme 1, remember these plants are increased in biomass. That biomass has to come from somewhere. We've shown that we can demonstrate that there's clear upregulation of certain photosynthetic genes, certain rubisco subunits and photosystem 1 components are markedly upregulated in the transgenic plants. So this is an interesting mechanism which may have implications to understand how we can manipulate biomass in not only canola but other crops as well. Next slide, please. We're currently conducting field trials, 2 field trials in Ontario, one in New Liskeard. On the right there, you can see this rather empty field, which was taken 10 or 11 days ago. And on the left, a field trial which is being conducted closer to home in Rockwood in Ontario, where the plants are growing very well. So we'll have a lot more to show, I think, this time next year when we've got the results of the field trials that are undergoing with these transgenic plants. The next slide, please. So there are a number of elements to this project. clearly understanding the biological mechanism in the green box at the bottom is of interest and importance to try and understand what we've actually done. The field trials are going to be important to hopefully demonstrate that we can maintain this increase in yield and biomass, and also the protection from environmental stresses in the field. So that's work ongoing. We're in the process of conducting further heat tolerance and drought experimental experiments, and also looking at root growth and root architecture in these plants. So far, we've only looked at the aerial parts of the plant. It would be interesting to also understand the potential for this increased stem phenotype, the increased diameter stem phenotype in the resilience to certain diseases like sclerotinia and clubroot. The increased cellulose, lignocellulose content is also something we want to look at in terms of the potential for reduction in pod shattering in these plants. The increased lignocellulose content of the siliques is something that we're going to look at as well. So there's a whole suite of increased agronomic traits or improved performance traits associated with these plants that we're in the process of examining and evaluating. Next slide, please. This is just to acknowledge, most importantly, Canola Council and the associated canola growers from the different provinces. Some other sources of funding as well, and in particular the Ontario Canola Growers Association, which are, and Ryan Koslag from the OCGA, I want to thank for his help in assisting us with field trials in Ontario. And we'd also like to thank Igor Falak from Corteva. Corteva have been great assistance with us for the field trial that's our local field trial in Rockton in Ontario. Okay, thank you.
Okay, so I'm Leon Kochian. I'm from the University of Saskatchewan. I'm the Canada Excellence Research Chair in Food Security here. I'm a molecular biologist, physiologist that works on transport and root traits and abiotic stress tolerance. So the title of the talk is Establishment and Application of a Brassica Tilling Resource for Development of Resilient Canola. Myself and Raju Datla have led this project, and we've got co-PIs from AAFC, NRC, UBC, GIFs, and NUSI. Kari Navavi is our industry partner with the field sites for us to do field testing ultimately. Okay, so why are we targeting canola? For one of the major themes of the agricultural policy framework, climate change, adaptation, resilience, sustainability. Well, that's obvious. I mean, Canada grows over 30% of the world's canola, very important to Canadian economy and agriculture. But also canola's crop performance is significantly impacted by certain aspects of climate change, specifically high temperature and drought. And we saw that in 2021 when we had a severe drought and there was a 30% loss in canola yield. So this project is centered around kind of the use of a tilling diversity germplasm resource, a tilling population, which will help to advance the discovery of genes underneath traits that we're interested in for this project, but of course can be used for any traits. Initially, this tilling population, and I'll explain it more briefly in the next slide, was developed by George Hahn at UBC. It's the only publicly developed tilling population in canola. And when he retired, he passed it on to Raju Datla to get it going. And it was funded by a Genome Canada grant. And one of the goals of that grant was to generate this resource in canola, make it available for researchers in canola in Canada, I think particularly Western Canada, but all of Canada. Now, we're interested in the climate change related traits, and that includes drought And we're particularly interested in water use efficiency because that intimately ties together drought with photosynthetic performance, which is, we can discuss this in the discussion session, often are at cross purposes, but we have found ways to be able to increase both of these aspects of the trait to increase water use efficiency, which is the unit of biomass produced per unit of water transpired through the plant. So doing this to develop climate adaptive canola. So developing this, get rid of this thing. Huh. Doesn't want to move. Okay. There.
We can see your mouse too, Leon.
Yeah. Okay. There we go. It was just blocking part of my slide. Okay. So when we talk about a tilling resource, I'll explain it very briefly. Obviously, genetic diversity is the cornerstone critical for advancing canola breeding or any breeding. And one of the ways that we can increase diversity is by mutagenesis. And tilling stands for targeted induced local lesions in genomes. And it's not a new technology. It was developed actually initially in 2000. And it involves mutagenesis, usually through EMS, so we can generate a series of SNPs across different lines of canola and look for changes. We can do both forward and reverse genetics. Forward genetics, looking for change in a phenotype of interest and then work back to the gene, or we can target a gene and look for allelic variation with different SNPs in the coding region or regulatory regions. So it's a technology that really was ahead of its time in terms of having the reference genomes and the sequencing and all the genomic resources. We can get a lot more out of it now. And this one, as I said, was developed by George Hahn, and Raju tells me that Erin Gilchrist is here in the audience, and she worked a lot on this population for George in his lab. So welcome, Erin. And it was developed in the doubled haploid 12075 genotype, which has a reference genome. Of course, now we have a number of reference genomes in canola, but this is the only publicly generated tilling resource for canola. Okay, so in terms of the genomic resources that we're generating for this, we're getting more bang for our buck by as typically done. Even though sequencing has gotten a lot cheaper, it's not cheap enough to do reference genome. Well, if you have unlimited money, reference genome sequencing on 2,000 to 3,000 tilling lines. So we're using what Raju developed, a three-dimensional pooling strategy where we can get good coverage with a much smaller number of sequencing runs. And on the reverse genetic side of things, we can identify superior alleles for a target gene. Or so like I'll talk here about our target of rapamycin kinase that we've been working with for a number of years, where we can design gene-specific amplicons for identification of allelic variation, specific gene variants of a particular gene in the tilling lines. And that is useful, I'll talk later about in terms of doing crop improvement through a marker-assisted breeding. And then, the phenotyping involves identification and characterization of lines, displaying in our project traits associated with climate change, adaptation, and resiliency, so drought resistance, and improved photosynthetic performance, not only under drought, but also overall, because obviously that could increase, it will have a capacity to increase yields under well-watered conditions. So in terms of the phenotyping of these tilling lines, we have several layers of phenotyping. We're phenotyping for yield or performance associated traits. So as we're doing the seed increases to redevelop this tilling line, we're also doing our initial phenotyping, in this case of shoot growth, shoot biomass, shoot architecture, also flowering and flowering time, and seed development. we're studying physiological traits associated with both drought and a kind of a connected photosynthetic efficiency. And these include measuring transpirational water loss, stomatal conductance to water out and CO2 in, and in terms of photosynthesis, chlorophyll content for better energy capture, and photosynthetic CO2 efficiency using a Lycor system. And so we're focusing on the identification and functional characterization of tilling lines that display the desirable climate adaptive traits associated with, the traits we're interested in: drought resistance, water use efficiency, photosynthetic efficiency, but also performance and yield. And of course, to really, drill down, we need to identify the genes and genetic determinants conferring these traits in the tilling lines in order to develop advanced breeding lines. So, as we do more detailed characterization of promising lines that came from the initial phenotyping that carry traits relevant to improving climate change adaptation and resiliency in canola, we'll use an experimental and functional validation of all these gene variants. So this particularly is useful, so you can start with a phenotype and work back to the genes, and also then, or work with a targeted gene. And either way, you can look at allelic variation within that gene to identify allelic variants that have improved performance for your traits. So that sets the stage for developing advanced breeding lines through marker-assisted breeding. And the application of this research, the research findings from this project, will allow us to develop advanced breeding lines for canola that have improved climate change adaptation and resiliency. And there's other spin-offs from this, as I'll show you, including improved nutrient acquisition efficiency. I'm not going to show data for that. So we've currently established through seed increase and phenotyping 1,000 tilling lines. We're finishing the second 1,000 this year. And we've performed phenotypic and physiological analyses and identified tilling lines displaying diversity in seed size in Raju's lab, and seed composition, particularly oil composition, some promising tilling variants from Mark Smith and Pengao at AAFC. Alterations in stomatal conductance and photosynthetic efficiency that increase water use efficiency. Now, in terms of a targeted gene, I'm going to focus briefly on our target of rapamycin kinase. It is the master regulator of regulators. It's been widely studied in human systems and in model systems like yeast. In recent years, a lot of study in plants, and it's a kind of a regulator of regulators. It regulates nutrient inputs and ties them to growth and development, or with nutrient limitations to reducing growth, possibly increasing programmed cell death. And so I'll highlight some of the findings in the next few slides. So here we show a picture of us. We phenotype, we grow 5 replicates of each tilling line. And while we're doing that, we phenotype them, in this case, for variation in leaf size, also flowering time, seed yield. I do a lot of root imaging at 2 and 3 dimensions. We have a nice high throughput root phenotyping system that allows us to phenotype thousands of roots on thousands of plants. And here you can see, a larger rooted and smaller rooted canola lines. And we're finding that the larger rooted lines do better under drought, often because they can acquire water at depth. But they also, it may have a win-win. I often will do better with lower NPK inputs. So there's really a very positive win-win situation. And of course, studying them for seed yield, nutrient uptake as a side thing with the roots, and drought resistance. This is just an example of work that was done by a couple of postdocs in my lab and more primarily Jura's lab on seed size. So here's the wild type, the DH102, 12075, and these are 5 tilling lines that have significantly larger seeds. So the 1,000 kernel weight shown here, it's about a 60% increase over the over the wild type. And this is seed area as a measure of seed size. And there's about a 50% increase in these 5 tilling lines compared with the wild type. And most of that increased seed size is due to increase in the embryo and also from the maternal side, maternal tissues, a greater, more thicker seed coat. So now I'm going to talk briefly about TOR as our candidate gene. Here we have the DH12075 and then the same line, dual haploid line, expressing high levels of TOR from different promoters. We've done this in transgenic Arabidopsis, in wheat, and now in canola, and in all cases we get increased drought resistance and water use efficiency and increased photosynthesis. But this is just well watered, and you can see the shoots are larger when you have higher TOR expression. The roots are larger when you compare the high TOR lines compared with the wild type. And the leaves, the shoots are larger because the leaves are larger, and this is simply the quantitative data validating that. We also see a quicker flowering time. So these plants have a shorter growth cycle, which may be advantageous under certain growing conditions. And then just finally, a quick snapshot of the increased drought stress resilience when you compare the wild type with the high expression TOR lines. This is well watered, where you can see the high expressing TOR lines of larger shoots. After 8 days of drought stress, they're all feeling the drought, but the wild type is much more drought inhibited. And then when we rewater, and 2 weeks after rewatering, the TOR expressing lines show full recovery. The wild type is stunted, does not produce much seed, and these all produce considerably more seed. I don't have the data for that here. So the project outcomes, the benefits to growers in the canola industry, we know we can produce, identify lines with improved water use efficiency and drought resilience. And this certainly benefits climate change mitigation and resiliency and yield protection against drought. The improved photosynthetic efficiency increases water use efficiency, but it also produces more carbon for increased seed yield, more soil carbon, and a lot of the carbon that's fixed in the soil is actually root exudates from the roots, okay? Low molecular organic root exudates, and that can improve soil health, presumably improve the root microbiome. You know, we'll be generating climate adaptive and pre-breeding canola lines, and the benefit, of course, is to develop improved canola varieties with all the economic benefits to canola industry and growers and stakeholders. Reducing greenhouse gas emissions, sequestering more CO2 in the soil through the larger root systems that have more root exudation. Reduced carbon footprint, particularly if we can show that they have reduced nitrogen needs, because we use 1 to 1.5% of the world's oil to produce nitrogen fertilizer, huge carbon footprint. And so this is a tilling resource for gene discovery of any trait and benefits to the entire canola research community. Finish off, these are the PIs and co-PIs that have really participated in this project. Raj, you and I are leading. Mark Smith and Peng Gao from AAFC. Daoquan Zhang, who is a Brassica genomicist from NRC. Of course, George Han, who is our advisor, you know, and has, you know, developed the first tilling population. And Katie Navabi from New Seed, who's our industry collaborator or co-PI, and provides field sites for us. And we have a group of really talented postdocs and research associates that have done a lot of the work in my lab and Raju's lab. And finally, I want to thank the Canola AgriScience cluster under the Sustainable Canadian Agricultural Partnerships in the Canola Council of Canada. Thank you.
I'll get us started here. My name is Gary Peng with the Agri-Food Canada in Saskatoon. The project is more about the development of a club root resistance and associated the application, especially deployment of the resistance. In the team, we have several co-investigators with a range of expertise from genetics, molecular biology, all the way to the canola breeding. And I'll jump right into the presentation. Next slide. See if I can move it. Yeah, it worked. Okay, a little bit of background of why we're doing this, and I believe that everybody is aware of the long-term threat of clubroot to no industry, and the challenge is that the pathogen population is highly diverse, and with more than 50 pathotypes identified, and that the residue spores, even portion of it can last for a long time in the soil. It's a hard one to be eradicated. Genetic resistance has been working reasonably well for us, and we've had the Generation 1 and now Generation 2 varieties available. But also, as we have observed, the resistance genes are relatively there are only limited number of our genes being utilized, and there's a risk of resistance erosion. And also, the point I want to make here is, resistance doesn't work in a vacuum, and not just about the genes. It's the additional management tools that we can utilize and to best deploy those resistant genes, and to achieve the strategy for the durable resistance. Okay, and with that, the backdrop, the main objectives of this the study, and it was to identify and develop unique or novel resistance sources. Over the years, different groups have accumulated quite a rich clubroot resistance resources. I will mention some of those, including both A, B, and C genome materials, and also we wanted to look at the resistance mechanism of those R genes, and in the single and the combination scenarios, and overall, we wanted to improve the diversity of the R genes to our industry, and also look at the optimized resistance deployment strategy, and to increase resistance resilience, and also, because of the breeders we have in the team, we wanted to even introgress some of the novel CR genes right into either the canola germplasm for the breeding industry, or even produce the cultivars directly. The methodologies we're using, I'll just kind of outline some of the key components, because depending on the project, that different methods has been used, all the way from gene discovery and the development all the way to the variety development. I think the resource development is a key part of this, including both A, B, or especially A, C genome, the CR genes, and also the tilling mutants, as mentioned by the earlier speaker, and the different molecular and the genomic tools are used, including the RNA-seq, genotyping by sequencing, and deep sequencing, some marker development, and even the CRISPR, the genomic editing tools, and to be used. We also use our protocols, look at the resilience of the certain genes or gene combinations, and also the conventional breeding techniques. What I want to emphasize here is all those technologies have been used in the different labs, so they are well established from our research work. Okay, because a lot of information from the different labs here, I'm just highlighting a few key progresses over the last couple of years. And this is a work done at the Dr. Chen Yu's lab at the AAFC Saskatoon. See if I can use the well, just I'll use the cursor here. Over the years, she has developed a large number of CR gene candidates, and gradually she has been focusing on those seven resource sources of the resistance. And in this particular part of the study, she sequenced all these seven sources, and in comparison to the a DH line without the resistance in it. So overall, this is the overview of the sequence data, but the key finding here is the number of the CR genes in those materials she has identified, and those are the key CR genes coming are being or have been developed in Chen's lab. Some of those have been transferred to the industry for resistance breeding, but she has been gradually focusing on a couple of more candidates with some unique features of the resistance. Here is the overview of the sequence data, and she was particularly interested in the ECD10 and this particular line of B napus, and it seem to carry a large number of potential CR genes, and it specifically, and she has this RCR9 and the 10 in mind for the development. And she also used the RNA sequencing analysis and characterized the different categories of potential disease-resistant genes in these materials, and in particular for the ECD10 and the line of B napus. And she has found a large number of potential disease-resistant genes in those materials under these the categories. So we'll see what will come out eventually come out from there. Well, this is just an example of how she further characterized those resistant genes in the material. Again, this is RCR10 and RCR9. And she has been working on, and this Manhattan plot shows the location of those genes on the different chromosomes. And the further she has been developing the markers and those SNP markers and corresponding to, for example, this RCR10. And versus the resistant susceptible line, and she is finding some novel features or even novel genes in this particular material. And another part of the resource development is the tilling lines. As previous speaker mentioned, we also looked into these mutants for potential club resistance or sources of it. And I'll just to mention that we've only screened about 300 of them. In those cases, the mechanism we hypothesis could be different because of those mutation, more likely interrupted the susceptibility related genes. And in this case, we did find quite a number of promising mutants, and for resistance to 3H, which may not be as big deal, but this is the original canola line, and this is a 2, 3. I couldn't even see it, probably 3A and 3D. But more importantly, this is the resistance to 9E, which is quite a virulent petatype against a range of different genes that we've tested. So we even found the mutants with the resistance to the 9E. So this aspect of the work, we wanted to seek the additional resources to fast track and to develop the new clubroot resistance sources. There are a range of work that's being done by Kay at U of A, and I did a little bit of that. Most of those work has been trying to identify the resistance mechanism associated with the different genes, and those work has been in progress, and some of the work has been published. So I'm not going to go into much of the details with it, but overall things are going well in terms of a number of the transformants being produced for the CRISPR knockout, and also for overexpression of certain targeted genes. There are a large number of candidates being looked at. And some of the work has been published in our lab. We look at the generation 1 and versus gene 2, resistance to 3H, and also to 5X. It looks like, in both cases, those resistance materials seem to induce the lignin increase in the cell wall, and for the resistance at the same time for against the 5X. And it seems that the similar pathways are being activated, but for the gene 2 varieties, there's a strong indication that those related R genes or pathways being more enhanced. So that's a little bit interesting, but the work is still being continued. This is work for the C-genome CR development, and Dr. Raymond used the resistance source from kale, and apparently the resistance in the C-genome is a bit more complicated, but he used the self-pollination up to the F11 stage, and I was able to find through rigorous selection, and he has found a good number of candidates with the canola quality. And as shown here, the content all the way to the seed quality. And more importantly, 6 of the F12 generation candidates selected, and showed a strong some of them, 5 of them, I believe, showed a strong resistance to 10 pathogen petatypes. That's quite a nice progress in his part. He is on his way to develop those into the germplasm for tech transfer. And he's also working on the mapping of of the C genome gene and try to develop effective markers for the breeding purpose.
Hi, Gary.
Yeah.
I'm just going to let you know that you have a 2-minute warning.
Okay, sure. Yeah.
Great, thanks.
Yeah. This is the last part of it. Rob Duncan has been using CR genes produced by Fenchain and to produce canola. I'll try to produce canola varieties, including the specialty varieties, and with the resistance. And the progress has been quite nice, and a lot of embryos has been collected, and including both from male and female gene pool. And there are currently about 700 plants are being harvested for the further development, either as a hybrid or the further crossing. So that part of the work is going well. And the last part is the resistance and resilience we're testing. Some of the work has been published. I think that the key point here I wanted to make is that the for the 3H common petatypes and the amount of soil inoculum makes a difference in terms of the resilience of the resistance, and it seems that the stack genes are better than a single gene, and we're testing the further the complex pathotype situation here, and we haven't quite finished. But that part of the work I will be reporting probably further in the future. Okay, just to sum up, and what all this mean to our growers, and I think we will have very good resistant resource being developed, and at the same time have a better understanding of this resistance, and we will have the CR pre-breeding materials being produced, and at the same time some of the even commercial varieties can come out from this program to support our growers' effort in club root management. And this is the acknowledgement beside the funding agencies, the grower groups, the breeding companies have been really supportive to the work by providing their resistance material for testing. So with that, I'll hand it over to you, Chris.
Hello, everybody. My name is Hussein Borhan. I'm with Agriculture and Agri-Food Canada in Saskatoon. The project that I'm talking about today is basically about protecting canola against blackleg by introducing genes and developing origin-specific markers. I'm the PI, and Nick Larkin at Canada Saskatoon is the co-PI. Collaborators are Ralph Lang and also Harsh Ramon from Australia. Let me go to the next slide. Blackleg can start infection at a very early stage, and so it affects the seedling, and the main disease really impact is at the adult plant stage. You see at the middle panel the cross section of the lesion at the base of the stem, and the far right is the extreme phenotype of resistant and susceptible. That the susceptible plant is almost dead, and the resistance that is basically in this case race-specific resistant could also protect plants through its lifespan. So genetic resistance is very important, and for blackleg, especially because there is no fungicide, or fungicides are not as effective as it should be, and it's the most cost-effective approach as well for any disease. So where the race-specific resistance, they detect the pathogen at the very early stage. On the left panel is what is the historic gene-for-gene hypothesis. It means that resistant genes, they detect their counterpart avirulence genes from the pathogen. If the avirulence genes is present and the resistant genes present, then we have a resistant phenotype. If either of those two sides are missing, meaning that we don't have a resistant genes, or our pathogen is virulent, meaning that it doesn't have the avirulent genes to be detected, then we have susceptibility. The right panel shows a very good examples of blackleg. Here is the blackleg with RLM7 resistant genes. Sorry, is this canola with RLM7 resistant genes. On the left side, you see that the left cotyledon has been inoculated with a isolate that doesn't have a AVR-LM7, and so we see a lesion here. On the right side, the cotyledon is inoculated with an isolate that has got AVR-LM7 and a virulent isolate, and we see this, the resistant phenotype. So I don't know. Sorry, stop. We have mapped about 11 blackleg R genes and identified some new and cloned 7 of the resistant genes so far. The latest of the resistant genes that we have through collaboration with industry are the LEPR6 and LEPR5, the two and here on the basically highlighted with the green box. These are a broad spectrum resistant genes that we identified a couple of years ago. The object of the project is cloning the LEPR1, LEPR2, LEPR5, and LEPR6, developing gene-specific marker for these genes, and also through collaboration with Harrow in Australia, that they have identified 2 new resistant genes called RLM12 and RLM13. The intention is to bring those RLM13 and RLM12 into Topaz background, a cultivar that we use this for integration of all of our R gene. That's Topaz is susceptible by itself. So Harsh Rahman will be doing that part of the work in his lab in Australia. Why we are cloning the R genes? Again, here we have the basically on the left side, we have that gene for gene chart. And as I mentioned, on the right side, we have the AVR and R genes that based on genetic analysis that on the pathogen and on the plant side has been going on for the last 2 decades. We have a very good picture of really the gene for gene complexity, this recognition complexity in blackleg, and probably is blackleg and canola is one of the most advanced in the crop environment. Actually, among the crops is the most advanced in terms of its genetics that has been defined. Groups in INRA has identified many of these AVR genes, and we have also identified a couple of them, including AVR-LM59, AVR-LM2, and we have, as mentioned, mapped most of these genes and cloned several of them. The one that are shown in red, we have the one that we have already cloned, and there are others, one that basically 12 and 13 that hopefully we will be bringing into the material through this project. And as you see, the recognition is not a simple gene for gene, but we have a really case of very complex interaction. For example, AVR-LM1 is recognized by both RLM1 and LEPR3, or in the case of AVR-LM47, it masks the recognition of AVR-LM3. So basically, if you have AVR-LM7, if your isolate is virulent, meaning that it has AVR-LM3, still RLM3 won't be functional. And so these are examples of this complex recognition. Why we want to, here is another reason that why we need cloning of resistant genes and how it could help us. In the past, we have, my lab has developed molecular markers, cast markers for pathotyping or determining the race of blackleg in the field. These markers has been based on they are allele specific based on the published avirulence gene sequence, and these markers have been given to the several diagnostic lab now, or they're offering it as a service. So as the other side of the coin, we need to know the origin genotype in our varieties as well, because knowing the races of blackleg and what sort of races are in our field will be not much of use if we don't know what sort of variety we have to use. And those variety should be the right type of origin to match that AV origin. And so in the right, we show that if we have the matching resistant against the AV virulence genes, we have the resistant reaction, otherwise we have the susceptibility. So the reason that we are cloning these genes is to help the farmers to basically have the tools for to identify the resistance in their collection, in their varieties, and use the right variety for their isolating their field. So here is the cloning step. We start with genome sequencing, then genome assembly, and then we identify the target genes. We put it in the right vectors, and we do transformation of the plants to confirm that we have the right gene. And if we have the right gene, then we go through this sequence and develop allele-specific markers. We have done Oxford Nanotechnology is a long-read sequencing that was done. That was achieved. We have sequenced LEPR1, LEPR2, LEPR5, and LEPR6. We have generated the draft genome. This table shows the genome statistics, that how basically the contic length and also the genome length. All of the genomes are around 1 gigabit. That is the expected size for Brassica and Naples genome. The bottom panel shows that the genome, we actually have a genome sentinel viewer, shows the part of the genome, and at the bottom we shows the predicted genes. So these are genes that are predicted based on computation and software, so that these are just computational. It's not the fact that these genes are correct genes or not, but this is basically using the RNA-seq evidence and other evidence, the computer tries to come up with the best possible genes that it's identified within it. So the next step is to find where is the integration line. We have marker for it as well, but in here we also took advantage of our genome sequence, and we use a SNP. We compare the SNP, genome-wide SNP in our sequence genome between the susceptible and the resistant lines, and we identified the SNP hotspot. So a SNP hotspot, we delimit the integration part. So these are topaz LEPR1, meaning the topaz is basically, as I mentioned, is susceptible. LEPR1 has been integrated into it by repetitive crossing. And topaz LEPR2 is again the same background. Topaz LEPR2 has been integrated by repetitive crossing. So here, when we compare these SNPs, we see that the SNPs are basically the hotstop is here A2, where is the LEPR1 genome is located, and another hotspot is A10, where LEPR2 is expected. So we identified the interval. Here is the alignment of contigs from LEPR1 genome against our reference genome. And this is a screenshot of the LEPR1 interval, not the whole part, but most of it. The bottom panel is the LEPR2 interval, just to give you an idea. These are all contigs that are aligned with the reference genome. Because with contig, we don't know which chromosome they belong to. We know where is our gene. We know it is on A2 or A10. So we have to align our contigs, see that which contig are aligned to A2 and which contig are aligned to A10, and focus on those contigs as potential interval for our genes. We then use RNA sequencing genes to go and annotate, correct the annotation, because the annotations by computation is not necessarily correct. For example, here you see that here is one exon that the gene is predicted, but actually clearly our RNA-seq shows that there is a second exon here. Also, we use RNA-seq data based for expression of the genes. The genes that are not expressed, we are not worried about it. The genes that are expressed would be of interest. We look at the gene function. We will look at genes that are similar to homology with our genes. So we target those first. And these are examples. Basically, this is the way that we go and narrow down so many genes within this interval to identify the most possible candidate genes based on gene expression, and it was based on the predicted function. Here on the top panel is actually LEPR1 candidate. Here is one of the LEPR1 candidate, and the bottom panel is one of the LEPR2. And if you see, actually, these are during the infection, and you can see that their expression is quite high compared to the neighboring genes. And the expression also, we look at the topaz expression, our susceptible line, and our integration line. And that's another indication to show that the gene, if the gene is not expressed in topaz and in resistant, then there's another good indication. We also look at gene variation. If the sequence are exactly the same and expressed in both susceptible and resistant, we will eliminate those because those are obviously not the right candidate. Going through this long process of is all manually basically annotated. We have identified 6 genes as candidate for LEPR1 and 2 for LEPR2. We are in the cloning process. We use a binary vector and we clone it into binary vector, and then from there we do the transformation. So the cloning is in progress at the moment. We took the same approach for LEPR5 and LEPR6. Unfortunately, our LEPR5 and LEPR6, because the LEPR5 and LEPR6 genome, both of them are at the end of the chromosome. So it is an area with highly repetitive area. Usually it grows the telomere. And we didn't have a good representation of context for LEPR5 and LEPR6. So we went back and did another round of sequencing. This time we used PacBio instead of ONT, Oxford Nanotechnology, and we have improved genomes. So here, if you see that ONT is the previous genome that for LEPR1, LEPR5, and LEPR6. It was about 900 megabase, but now it's over 1,000 megabase. And our context also has improved in terms of its length. So we just got these two new improved genome, and we will be looking into taking the same approach to identify LEPR5 and LEPR6 candidate. As I mentioned, we have been working with our Australian colleague. They have identified 2 genes, one of them RLM12 and RLM13. We had a very difficult time for signing the MTA. Basically, we signed the MTA with Dr. Harsh Lamon's lab and their institute, but later on, it was higher level of the Australian government stepped in and they delayed the process. I think it was at the even foreign ministry level. For 1 year, at least, we couldn't move forward with this part of the project, but that obstacle has been resolved now. And Dr. Ramon recently has started working on this material and received seeds of topaz from us. Hopefully, we will integrate these 2 genes into the topaz and provide it to the Canadian canola growers. As a summary, so we have sequenced the genome of topaz LEPR1, topaz LEPR2, LEPR5, and LEPR6. We have assembled annotation of these genomes. It's completed that part. We have done additional genome sequencing for LEPR5 and LEPR6. And we have also identified candidate for LEPR1, LEPR2 that hopefully by the end of summer, we will be able to move it to the stage of transformation. And the work on integration of RLM12 and RLM13 has just started and by Dr. Ramon in Australia. I would like to thank people in my lab that have contributed, especially Elena Bendon, a technician in my lab, and Anna Montenegro, a postdoctoral lab in my lab. Isabel Parkin, Kevin Coe, Aaron Bollett, and Venkat Bendy, and Sampad and Andrew Sharp are from GIFS and AAFC that have been contributing to this project. Aaron Higgins from Isabel's lab has been really helping us with DNA extraction, high molecular weight DNA extraction, and some of the making the library for the genome sequencing. Also, I would like to thanks the founder of the project and basically grower organization and Ag Canada as well. And also thank you for your attention.
So in the first presentation here, I'll be speaking about fungicide sensitivity in Sclerotinia sclerotiorum. I'm actually speaking on behalf of 2 of my colleagues who've done more than the lion's share of the work on this project, Dr. Lona Buchwald who is responsible for collections. And given all the, I guess, the difficulties in accessing land these days, it's a monumental task, and she does a very good job. And Dr. Edis Zianovic, who is a biologist working on the project, and he's responsible for sample preparation, testing, and genetic analysis, and I'm speaking on their behalf this morning. So Sclerotinia is the causative agent of a disease called stem rot. On the left here, you can see what becomes of canola if it's heavily infected. Last year was a very bad year, and we would have seen, many fields that were affected to this extent. Management of Sclerotinia is primarily through, I guess, three means: crop rotation, which is important for all pathogens. There are a few tolerant varieties, and I spent enough time crawling around on my hands and knees on my own land and in neighbors' fields last year to know that these actually hold up quite well. And then there, of course, is fungicide application, which is part of the package. It's important going forward, though, that we prolong the usefulness of these fungicide active ingredients, which requires rotating between fungicides with different modes of action, using fungicide mixtures, which are becoming more common now in the new products that are coming out, and also to apply these fungicides only when necessary. And that's aided by the various risk management assessment tools that Lona and other people have been developing. Before I go further, I just want to make a note of the funding. The funding for this project initially was from the Canola AgriScience Cluster, or SCAP, where we received funding to test 3 fungicides from 3 different groups: prothioconazole, boscalid, and azoxystrobin. And I'll be presenting that research here today, or at least the initial findings. Based on those initial findings and some interesting bits within, we were able to obtain some additional funding through CARP to support the testing of 3 additional fungicides, 2 other Group 7 fungicides with slightly different chemistries, as well as another Group 12 fungicide that appeared on the market in 2024. So the work plan is to collect isolates from Alberta, Saskatchewan, and Manitoba. We've been doing that through the 2023 through to the next year, through 2025 growing season. Compare these to a collection that Lona had put together in 2010 to see whether or not there's been any differences in fungicide insensitivity. We're testing against 6 fungicides. The work is actually being done by a third-party contractor, Epilogic, and they do all of the testing for the European Union. We retest the most insensitive isolates just to verify them in Saskatoon using the same method. And then we characterize mutations and enzymes and pathways for any isolates that we notice that have increased insensitivity or resistance, and then develop DNA markers linked to these mutations to make testing that much easier going forward. So this is a provincial map of the collection sites. This was from 2023, 2024. This is just most of them. By the time we get to 2025, there'll be hundreds of sites, but we're collecting throughout the entire prairie canola growing region, all the way from well up in the Peace River country to down in the southern reaches of Manitoba. This is only possible through sort of friends and collaborations in Alberta, both at the provincial and the federal level. Mike Harding, Greg Daniels, and Kelly Turkington. In Saskatchewan, through our friends at Richard the Pioneer. And in Manitoba, through Sonia Wilson and our very own Chris Mancher, who provided some of the collections in Manitoba. So the fungicide testing itself is done using a microplate assay. This is a protocol that follows the Fungicide Resistant Action Committee guidelines. And so it's a standardized protocol. I guess importantly, the fungus is grown in these microtiter plates at increasing concentrations. At some point, the growth diminishes, and we can use this data to calculate a very important parameter called the EC50, or the effective concentration 50%, which is the amount of fungicide that inhibits the growth by about half. I want to make note of the controls. So for every fungicide, Epilogic provides a sensitive, adapted, and in some cases a resistant isolate. And it's important to differentiate these. So obviously a sensitive isolate is sensitive. An adapted isolate is an isolate that has undergone small changes that allow it to increase, to survive higher levels of the fungicide. These could be changes that might interfere with the uptake of the fungicide or the activation of systems that would pump out the fungicide from the cell. but they can have increased tolerance to the fungicide, and this can have ramifications for field efficacy. And then there are the resistant isolates, and these are isolates that have undergone a mutation in the protein that is the target for the fungicide. And these have hugely enhanced resistance to the fungicide, making the fungicide completely ineffective in field situations. So I'm going to show you the data from azoxystrobin, prothioconazole, and boscalid, and you'll see how these three classifications of sensitivity factor in. So here we have mapped out the data for the EC50 values from isolates from the 2010 collection and the isolates that we collected in 2023, 2024. And overall, the population was sensitive to azoxystrobin, which we found a bit surprising because azoxystrobin has been applied for at least 20 to 25 years for stem rot control. Although we did see some isolates with increased insensitivity or adaptation, and I hope my mouse is working here, in both 2010 and 2023, 2024. And there might have been a slight increase in insensitivity between the 2023, 2024 population compared to the 2010 population, but overall the population is sensitive to this fungicide. We see a little different situation with prothioconazole, in that if we look at the sensitivity of the range of isolates to both the sensitive and the adapted control, we can see that a large proportion of the population is in fact adapted or even highly adapted to prothioconazole already. And so we didn't observe any genuine resistance, but we can see that the population overall already has a higher degree of insensitivity to prothioconazole, so it's primed for that. We didn't, however, see an increase in insensitivity from 2010 to 2023, 2024, if you simply compare the blue and the orange lines. So the situation appears to be at least stable. In regard to boscalid, we see a different story again. So like azoxystrobin, the overall population in both 2010 and 2023-24 is sensitive to boscalid, although we did see some isolates with increased insensitivity, as we did with both of the other fungicides. So there's some adaptation occurring, or there are isolates with some adaptation. More importantly, though, we did find an isolate, and I'll show you where in a bit, that was genuinely resistant to boscalid. And as a matter of fact, we had to chart that on a different scale. The EC50 value here is orders of magnitude greater than what we see in the sensitive control. And this particular isolate, 2024 Manitoba 12 cm, is about 100,000 times more resistant to boscalid than the sensitive control. As a matter of fact, it's even more resistant to boscalid than the resistant control that Epilogic provides in their testing, which is the mutation H146R. And so this is something we have to be genuinely concerned about. So we took it upon ourselves to actually sequence the target gene in this particular isolate. The target is succinate dehydrogenase B, and we found a mutation in that gene which changes a single amino acid from a proline to leucine at position 226. And this was actually very interesting because that mutation has been actually identified before as causing resistance to group 7 fungicides. In fact, that particular mutation is known to cause cross-resistance to a broad set of SDHI fungicides. And in fact, in 2022, work coming out of China identified that same substitution in a sclerotinia isolate and found that it had cross-resistance not only to boscalid, but to 7 other SDHI fungicides, carboxin, fluopyran, pidiflumentvin, flubenna-teram, pyrazivana-flumid, fluinidipyr, and penthopyrad. I practiced those last night, and for the life of me, I mean, I really can't spit them out properly, so it's a bit of a chore. But more importantly, two of the fungicides, the Flopyrum and the Flumentifin, are two of the fungicides that we'll be testing going forward with the CARP funding. And of course, we'll be testing for cross resistance between this particular isolate and these two other fungicides. And we expect that there will be cross resistance. These two fungicides appeared in some of the newer products that are just coming out for Sclerotinia control. And it appears that we may already have cross resistance in the population. In case there are any farmer friends online who may be interested, this particular isolate was collected in Westbourne, Manitoba. I think it was actually collected by Chris, and we'll be following up on this. So in summary, and I don't know where I'm at for time, I'm trying to go through this a bit, maybe too quickly, that the overall insensitivity to prothioconazole, azoxystrobin, or boscalid did not increase from 2010 to 2024. So the situation, at least in the field, appears stable. However, we did find isolates with elevated insensitivity or adaptation to all three fungicides. I think if we had to make any recommendations early on in the project, we would say that we want to exercise caution regarding prothioconazole. There's already a large fraction of the population with some level of adaptation to prothioconazole. Interestingly, the patent protection on prothioconazole expired, and this year we saw many generic products appearing that were registered for control of stem rot in canola. I think there are now 9 products available, of which 6 or 7 appeared last year containing only prothioconazole. So if the market could very well be flooded with cheap generic prothioconazole, which could lead to its overuse and which could further exacerbate the situation. So I think this is something we want to continue to monitor. Also, caution regarding boscalid. We did find a highly resistant isolate in Westbourne, Manitoba that has a mutation that will likely make it cross-resistant to either group 7 fungicides. If you look at the map of Europe, in particular France, it is littered with Sclerotinia isolates that have resistance to boscalid. So what's happened in Europe, I mean, it can happen here as well, and I think we want to be careful that it doesn't get away. Our focus, as a matter of fact, next year will be more so on Manitoba, and we'll be doing a lot of collecting there. And we'll be testing all 6 fungicides. These represent almost all the groups and chemistries registered for Sclerotinia control in canola. These would have represented all the groups and chemistries in 2024. There's actually one new product that's been registered in 2025, which is a group 21 fungicide, and most of the new fungicides that are being registered are usually contain 2 components. So the industry is beginning to address this this issue as well. So I guess thank you for your attention. I believe both Edis and Lona are online, and so they'll be able to participate in the Q&A afterward. And again, I'd like to acknowledge the funding from the Saskatchewan Canadian Agricultural Partnership, the Government of Canada in this case, and the Alberta, Manitoba, and Saskatchewan Growers Association, both in their support of the SCAP as well as in their support of the CARP project to continue or expand the fungicide testing. So I'll now speak to another project which I'm also co-leading. This is addressing flea beetle resistance in canola. It's actually SCAP Activity 16. This project is actually a little further along than maybe some of the others because it's actually the second iteration of cluster funding. We had cluster funding in cluster 1 to support some of the initial work, and this is actually following that. I have 2 co-investigators in the project, Isabel Parkin and Sally Vail, but all of the work, I guess over the last 10 years, has been done by a research associate in my lab, Dr. Zora Hedarian, and she's really the expert on this. So some of you may be familiar with flea beetles, especially anybody trying to do field work or actively farming. I guess the image on the left is not unusual in canola fields that have been not treated, for flea beetle control. We have two flea beetles in Canada: the crucifer flea beetle that used to be the predominant species and sort of still is, depending on the area, as well as the striped flea beetle, which has been increasing in abundance and has become of particular concern. So flea beetle control in Canada. So there's no resistance available to flea beetles in any canola varieties that I am aware of. Okay. Since about 2001, all canola seed in Western Canada is treated with a Group 4A neonicotinoid insecticide. These can be things like Helix or Prosper. So any commercial seed that you buy will be treated. However, you can also purchase additional seed treatments. These are generally targeted for some additional flea beetle control as well as cutworm control. And some of the more common ones are Buteo, which contains a Group 4D insecticide, Lumiderm, which contains a Group 28 insecticide, Fortenza, which actually contains both a Group 28 and a 4C insecticide. These are primarily for flea beetle control, but there's some carryover and they do control flea beetles a bit. However, despite all of this, issues with flea beetles remain. The striped flea beetles are less affected by the neonicotinoid insecticides, especially the Group A insecticides. That's now become well known. It's published, and that's leading to problems with the striped flea beetles. And often requires spraying of foliar insecticides. These are often older pyrethroids is sometimes required after the fact, just to keep them under control. So the approach that we're taking, and this has been talked about for quite a while, is to try to introduce hairs or trichomes on the canola leaves to prevent feeding. And I'm just going to play this little video of essentially we have two leaves, one hairy leaf on the left and a non-hairy leaf on the right. And you can see when the beetles cross the hairy leaf, they very quickly become uninterested and leave the leaf. And this is due to something that one of my colleagues, Dr. Julie Soroka, documented, whereby the beetles have to go through about 7 different steps in order for them to finally settle and begin feeding. And if you disrupt them at any one of these steps, they have to reset the whole program and go back to step 1. And this occurs actually fairly quickly. And you actually send them into this loop, if you want to call it that, and eventually they simply give up and move on. And I've made this analogy probably every time I talk to it. It's almost like a dog having to turn around 3 times before it settles on the pillow on the couch. And if you poke it at turn number 2, it'll have to go back and keep resetting. And so it's sort of a similar experience. So we're looking in this particular SCAP project at naturally hairy Brassica species, and my colleagues Margie Gruber and Julie Soroka screened about 1,000 Brassica lines from different plant collections around the world. And what they were able to identify were hairy lines, Brassica napus, Brassica rapa, as well as a Brassica villosa, which is related to the vegetable Brassica species, Odoraceae. I want to point out, though, that hairiness is rare, in particular in Brassica napus. So of the 300 lines they screened, they found only 12 that had hairs. The Brassica napus and Brassica rapa have these cactus-like hairs, whereas villosa has these very soft type of hairs that almost make the plant feel a bit like velvet. So the goals of the project are really twofold: to identify the trait for hairiness in Brassica napus, to identify the trait for hairiness in Brassica villosa, and to move these into contemporary canola varieties, or at least along that path. In order to make this work, we first have to understand something about the underlying genetics. And so to do this, we cross a hairy Brassica napus with a glabrous or non-hairy Brassica napus. We develop a progeny, in this case, that has slightly fewer hairs than the hairy parent. And then we develop a population from this progeny or F1 line. And then we look at that population and we try to associate the phenotype, which is hairiness, with the underlying genetics or genotype of these lines. So when we looked at the population, in this case, we looked at 300 lines within the population. These are phenotyped for hairs. We looked at the third and sixth leaf. We measured or counted the hairs on the top, bottom, petiole, and perimeter. This is all done under a dissecting microscope. 5 replicates per line. Poor Zora did 12,000 measurements. I really thought at some point she was going to quit, but she persevered. And indeed, when we correlated those measurements with the genetics, we were able to identify a major region, QTL, a quantitative trait locus, on chromosome 11 that explains hairiness on both the top and the bottom side, the perimeter, the petiole, and on the total leaf. So after 5 years of effort, this is what it yields, a peak on a graph, and you would have to be a scientist like myself to get very excited about that. But we looked at this particular region, and I think we've narrowed it down to a single candidate gene that we believe to be responsible for regulating hairiness in this Brassica napus line. So one interesting thing that fell out of this research was that when we looked at the population of lines that were derived from the hairy and the glabrous cross, we found many lines that actually had an enhanced phenotype. So the original DOS2 hairy parent had about 350 hairs per leaf. Within the population, we have lines that have over 800 hairs per leaf. And this is not uncommon in these types of mapping populations. It's a phenomenon called transgressive segregation, where you get traits from both parents. But what we've done here is that we now have true breeding lines that have at least 2.5 times the amount of hairs in the hairy parent. And we've really set the bar, much higher as to what the upward potential can be for leaf hairs in Brassica napus. So, of course, you're wondering, does this really have any effect? Well, in order to measure that, we first had to spend some time developing a better flea beetle damage assessment tool. This is the traditional method for examining flea beetle damage, where you simply compare pictures of your leaves and cotyledons to pictures of plants where somebody has determined how much leaf area has been eaten. But this wasn't a fine enough tool in order for us to differentiate, lines that might have subtle differences in flea beetle damage. And so we developed, spent quite a bit of time doing this, developing our own damage assessment tool. And we're using a piece of software that that we use for examining confocal microscope images. And it uses AI to train what the software can image. And so after the bioassay is done, we remove the leaves and the cotyledons. We take pictures of them, and then we use the software to calculate the entire surface area of the leaf and the cotyledon. And then we can train it to identify damage due to pits, which is where the beetles begin feeding, damage due to perforations, where the feeding has gone all the way through the leaf, and damage due to edge feeding. And then it will sum the total of all of these, both individually or in total, and we get a very accurate assessment of flea beetle feeding damage using this AI-guided tool so that even small differences can be noted. In applying this new bioassay system and these tools, we were able to establish that the hairy lines within the population and the hairy parent do indeed have reduced flea beetle activity and flea beetle feeding compared to the glabrous parent, which in this case is now 0. And damage was reduced here by anywhere from 25 to almost 50%. And so this really, confirmed in our minds that, hairs can in fact reduce damage to a fairly significant degree. It's not absolute, but the damage is clearly reduced. We did the same kind of mapping exercise in Brassica villosa, but in this case we identified not one but 3 regions that are responsible for trichome number or trichome abundance in Brassica villosa. 1 major QTL off to the right here, and then 2 minor QTLs. We also mapped a locus or a gene that is responsible for regulating trichome length. Now, when we did the similar bioassays with the material from the mapping population, We were clearly able to establish that the lines that had, that were very hairy and had long trichomes were as, resistant to the flea beetles as the hairy Brassica villosa had, some as little as, only 15% damage. Whereas the lines that had no hairs were clearly more damaged than had the highest, level of damage compared to the control. But we also saw a number of lines that were in the middle here, and we were kind of curious as to what's going on. Okay, and these lines that we called were mixed phenotypes. And so when we looked at these a little bit closer, we found that those lines not only vary for hair number, they also vary in hair distribution. So some of the lines have hairs only on the veins, some have only hairs on the perimeter, some have hairs only on the upper side of the leaf, but not on the lower side of the leaf, and vice versa. They also vary for hair length. So we actually have lines that are breeding true for small, medium, and large long trichomes. And interestingly, they also differ in two other traits that are known to be involved in insect resistance. One is the presence of leaf wax, and so that's been shown to be a deterrent for flea beetles to initiate feeding, and also a purple pigment that appears on the leaves. This is called anthocyanin. You can see it around the edge of the leaf here, and this is the same compound that you would see in purple cabbage, for example. And this is also known to deter flea beetle feeding. So there are actually multiple traits within Brassica villosa, that being hairs and hair length, leaf wax, and the presence of the anthocyanin pigment, all of which contribute in different ways to, to reducing flea beetle activity. So the conclusions or the outcomes here, Brassica napus with hairs that we've developed have reduced flea beetle feeding activity. In our laboratory bioassays, they reduce damage by about 40% on average. These are being distributed currently to both public and private sector breeding companies. We're hoping to begin testing some of the material in the field next year in studies that will be with and without insecticide treatment, sort of the ground truthing or the real world experiments. The second outcome is that we have Brassica villosa, Brassica oleracea lines with high levels of resistance. Some of these are hardly fed upon at all. And there are multiple layers, multiple types of resistance in these lines. They're both hairs and wax and pigment. We actually are undertaking some work to map the genes responsible for wax and pigmentation. And these have also been distributed to a public sector breeding organization. They'll be testing those in the field next year. Work or the challenges ahead, I just mentioned this, to test the Brassica napus lines in the field with and without seed treatments to see how well they hold up. We want to see whether or not we can further increase hair number in Brassica napus. As I said, we've increased the upward potential by about 2.5-fold. Are there ways in which we can increase that further? And so we've undertaken some experiments to do that. And finally, can we introduce any of these really interesting traits from Brassica villosa into Brassica napus? We've currently set out to do these wide crosses. It's challenging. We're making some headway, as any of the breeders would know. But we've spent quite a bit of time, seeing just how feasible this is. So all in all, I think the work is going actually quite well. We'll see how the lines hold up in the field over the next couple of years, and in the process, we'll be getting a better understanding of the trait, and hopefully along the way, developing even better material.
So feel free to questions in the Q&A, and unfortunately, it's like I was asking most of the questions, and in no particular order, but only Dwayne, and I've been following your flea beetle work for years. Trichomes on the petioles, we've identified that as maybe a weak point in the feeding habits or preferences of flea beetles when you challenge them with hairs on the leaves. At least one of your lines looks like it has some hairiness extending to those parts. Is that a goal of the program? Is that something you've already identified?
As a matter of fact, Keith, it was a goal, and the line that we had selected for mapping does in fact have hairs on the petiole. And we do look at, we've counted hairs on the petiole as well. The same gene that we've mapped in Brassica napus controlling leaf trichomes also controls petiole trichomes. And we also assess the damage on the petioles using the same AI tool.
Excellent.
So it seems to be part and parcel of the same trait, which is fortunate.
Absolutely. Insects seem to not enjoy starving. They adapt a little bit if you challenge them. Moving to the Sclerotinia presentation, the site in Manitoba that you identified as having some really high levels of resistance was that apparent to the grower, or was was this a surprise, or was it a problem field when it was collected?
Well, I might have to bring Chris in on that because I think Chris collected that isolate. No, I don't believe that, that the grower was aware of this. Of course, we are looking for any anecdotal evidence where growers might have had a failure in terms of fungicide application, and it would be nice to step in there and monitor those fields. I'm not even sure if the grower has been notified. This is all very recent data. We received it at the end of March. We shared it with the Sclerotinia steering committee, would have been in May. And I think we're working on a method and messaging going forward as to how do we communicate some of the findings we might have, at least early on, to growers when it could impact decisions going forward. Obviously, we want to do more testing either in this field or in this region this fall.
Yeah, I can add just a really brief note. This field that the isolate was collected from did have some significant Sclerotinia damage. It was in a little bit of a lower part of the region, I guess, in terms of topography, so it would be a little bit higher at risk, closer to some maybe higher moisture conditions, especially being near a river or creek. We haven't reached out to the grower yet, but working with the agronomists in the area, and we'll be doing so this summer and probably into the fall, having a better understanding of what are the management strategies involved for Sclerotinia in that area, and probably having that discussion to see if we need to think about this plan kind of a little bit more in depth.
Keith, we're really hoping this is just a one-off thing, but we don't know yet. So, I mean, ideally, I would like to find nothing, but I suspect that will not be the case. So,
No, not after your comments. France having issues. So yes. Question for Hussein. Growers are just, I think I could safely say growers are just finally wrapping their head around, the concept of races in blackleg, which is surprising because we've got that really well developed and understood by a lot of agronomists already. But they're wrapping their head around races, and we begin to understand that RM3 is really no longer present in Western Canada. So the genetics we've used for 20 or 30 years for blackleg resistance aren't working. But I'm not sure if I understand myself, but would the new genetic material, the 12s, the 13s, the 14s that you're discussing, are those resistance genes useful, or is that avirulence genes already present in our population? How does that work when we find new genetic resources? Are they deployed in the field and recognized as and can those genes be activated by the races present in?
Yeah. Sorry, can you hear me?
We can hear you, but we can't see
Yeah,
you. But
sure.
that's okay.
Yeah, I don't know what's my yeah, I think I'm trying to. Yeah. Anyway, so Ireland trees, very special case, as I mentioned in my presentation. Ireland trees not being effective is mainly because the presence of AVR LM47 that mask the recognition of avirulence genes. So that basically, it doesn't mean that the races doesn't have the avirulent genes that is needed to trigger the RLM3 recognition. It is the presence of AVR LM47, another AVR gene that mask that recognition. So it basically make RM3 ineffective. And RM3, based on the screening that we have done, is widely present within the basically probably 90% of the Brassica napus that we have from the germplasm, from the PGRC germplasm that we have looked. The only widely present is RLM3. That's the issue with RLM3, and the same thing with RLM9. Back to your question, majority of these genes, like we have already introduced, actually we have mapped and given the lines RLM11. Based on our evaluation, AVR11 races of blackleg that has got AVR11 are widely present again across Western Canada. So RLM11 should be quite effective. LEPAR5 and LEPAR6 that we have identified a couple of years ago, and now we have given it to the industry to use in the breeding program. Those are, we don't have, I think for LEPAR5, I think we have one or two isolate that are, one isolate that is virulent, but we don't have anything that if out of 30, 40 isolate that we take, that is being able to overcome those LEPAR5 or LEPAR6. And the same, we have shared those with our Australian colleagues that they haven't been able to find isolate that is basically virulent toward those lines. So I think we have enough genetic material within our selection, our collection, the one that we have given RLM1, 2, 3, 4, 7, and 9, 11, and the LEPAR1, LEPAR2, LEPAR3, LEPAR5, LEPAR6. So there's quite a few that all of them should be effective, except, as you mentioned, RLM3. So I think in terms of genetic material, we blackleg is in a good stage. I think we can really manage the disease with using, is really the how we manage it, is not that we don't have enough resistance. I think we should really monitor the races in the field, and we should now, with these markers that we are developing for our genes, we should have both tools for the farmers to use the right resistant genes for the isolate in the field.
Excellent, thank you. Looking at the questions that are coming in for Ian, do you have comparative seed composition data for oil, protein, fiber for the edited gene material versus the wild plants? You've indicated that oil composition doesn't change. I guess this question is coming in. How about the other components of the seed?
We're yet to test the protein, but that is something we have in the pipeline. We're going to test the protein content. That's obviously important, but it's not been done yet. But as far as oil quality is concerned, there's no alteration.
Excellent, thank you. And for Gary Ping, I had a question here. You have stacked some clubroot genes on a slide near the end of the presentation, and on the resilience slide, I believe it looked like the incidence of disease dropped into cycle 2, but then it seemed to come up and go back down. Is that simply a function of not enough cycles to see those resistance genes overcome? Is there some comment you can make on how successful we'll be stacking those genes, or is there a reason for that to go up and down?
Yeah, I was better running out of the time, so didn't have a lot of time to explain that fully. It was a bit of a continuation from our earlier work, looking at the single versus stack, and also the initial inoculum level in terms of the resilience of the resistance. In this case, we included some novel materials for the pathogen pathotypes. We have the mixture of those pathotypes representing the predominant ones, and also that the 9E as a segment of a potential highly virulent pathotype. And in the study, there's always a bit of a variability, but when you look at those curves are coming down quickly, and with for the resistance part, that was a Gen 2 variety, and plus a new variety with a Gen 2 plus with the 3 CR genes, and also a gene added at the line and with a knockout of a susceptibility factor. And those are the ones that seem to, and a couple of the ones like the Gen 2 variety had a very high initial inoculum and then start coming down quite quickly. So we needed to interpret that. In our case, we thought that the 9E with fairly small portion in the inoculum may be sufficient to cause the high amount of disease to start with, but then the inoculum putting back there may not be as large to be able to repeat that kind of the virulence. So that's coming down. The some other trend further down is a bit of a natural variability. They're not really significant in terms of the stats. It seems that the resistance has been maintained, and since the cycle 2, we're still completing the cycle 5. There's a bit of a issue, caution with that simulated testing is that the amount of inoculum we're putting back from the diseased plants is less than the field condition, because we're dealing with the full size of galls with a lot more inoculum. In this case, the plants were smaller. We harvested it after 4 weeks of inoculation. The galls are smaller, providing less amount of inoculum. So whatever portion is responsible for causing the disease and may not get amplified in terms of amount of inoculum in the next cycle. So that's probably the explanation of it. So the trend was not significantly going up. It's more of a kind of a maintain that level, and they're not that statistically significant.
All right, thank you, Gary. In the interest of maintaining our timeliness and actually letting people have a break for lunch when they're coming back relatively quickly here, I'll let Chris come on and remind us what time that is. But just a reminder for the speakers that we do have some questions in the chat that probably didn't get answered, and you can get to that. Already some folks are wondering if your material is available to look at, and Ian, they're suggesting that you have a commercial hybrid so we can see if your material is yielding okay in the 2 field trials. And I don't know if it's an appropriate joke to make, but your branch starch modifications really looked like you were just modifying branching on the plant, but that's simply I had to keep reminding myself that branched was a starch structure you were talking about, not a physiological, not a physical feature, but interesting presentations from all.
Yeah, I'll respond to that comment then online later. Sure. Excellent. Not about the branching, but about elite lines.
Thanks, Ellie, for the introduction. My name is Emmanuel. I work with the Integrated Agricultural Technology Center here at the Lethbridge Polytechnic, and I will be presenting to you what we at Lethbridge Polytechnic have been using the canola mill for in our research, and this is SCAP activity number 14. So as we all know, and to begin with, we all know that the world population is increasing and is expected to reach around 10 billion by 2050. Therefore, there is the demand for sustainable food production system in order to cater for the growing population need and provide food security for the people. In recent decades, aquaponics has gained significant attention and holds a considerable promise as one of the sustainable agricultural practices that can help provide food security to the growing population. It is a harmonious integration between aquaculture and hydroponics. So basically, this innovative system involves the cultivation of fish and plant in a symbiotic environment where the waste produced by the fish serves as a nutrient source for the plant, and in turn, the plant also helps purify the water, making it suitable for the fish in the recirculating system. In an aquaponic system, the primary source of nutrient is through the fish feed. And conventionally, this fish feed is the protein source of this fish feed is mainly from the fish meal. And this fish meal, however, is not sustainable. And because of it not being sustainable, there's the need to be able to look at innovative measures in order to be able to overcome these challenges. This fish meal, like I said, has been a major source of protein for these fish feeds. However, these fish feeds are at risk due to their increase in demand and also unsustainable practice, and as well as the high cost that they come with. And therefore, the need for a more sustainable protein sources in order to be able to produce those fish feeds. Plant protein sources have become the go-to now because of some of their significant attention that they've gained over the years. And canola meal has been one of the plant protein sources that has gained significant attention due to its high protein content and its ease of availability and low cost as well. And this is the solid matter that is left after extracting the canola oil from the seeds, and it's processed into the canola meal. This canola meal has been a suitable alternative as a plant protein source due to its ease of availability, high protein content, usually above 40% in its dry matter content, and also its low cost. So in this presentation, I will be presenting to you what we at Leadville Polytechnic have been using canola meal for. And here I'll be highlighting our latest research that we did using 33% of the canola meal on tilapia and lettuce in our system in order to be able to determine how they grow the fish and plant production system and overall water quality. I also highlight the nutrient allocation within the aquaponics system. Here is our aquaponics system that we have at Leadville Polytechnic. And here, as you can see, we have, these are 20 microsystems. And schematically, we have 3 plant trays which contains raft where the plants are held. Beneath that is a fish tank that holds the fish. And we also have a filtration tank which filters our waste, and it's also able to collect our sludge, and also a pump that pumps the water system into the system in order to be able to recirculate in the system. To begin with, we first grow our seedlings for about 21 days, and we make sure they get into a uniform state that we can be able to transfer them into our microsystems. Once the fish and plant goes into our systems, we feed the fish and plants. We feed the system with feed. The fish feeds for about 21 days for 3 weeks, and see how compare the growth rate between our control, which in this case are the fish without the canola meal, and our experimental units which are fed with the canola meal. After this, we now harvest our plant and fish separately. We dry them and get their weight content, and we analyze them we take them through various processes. First, we do our physical digestion. From there, we do our chemical digestion using our microwave. Then from there, we analyze them with our ICP instrument. Here are some of our results. First, I would like to talk about our water quality. Water quality play a critical role in maintaining the aquaponics system. And conventionally, these water quality parameters are measured with handheld probes. And these probes are labor intensive, and they're also prone to human errors and inadequate to address if there are some rapid changes within the system. So we came out with an innovative way to be able to address this. And this is by using our Raspberry Pi system, which is able to automatically monitor our data our parameters in the system, such as pH, temperature, dissolved oxygen, and electrical conductivity. These Raspberry systems have relays that are connected, are equipped with relays that are connected to peristaltic pumps that pumps or are able to dose our acid and base into the system based on our set parameter that we set, and is able to control the pH and other conditions within our system. First of all, this is the result that we get for pH. As we know, pH affects fish, plants, and also beneficial bacterial health within a system. And optimal pH helps to ensure that there's nutrient availability for plants and fish toxicity as well. As we can see on our system, we are on the graph, we are able to maintain the pH between 6 to about 6.7, which is the optimal pH range for our fish and plant in order to be able to have sufficient nutrient within the system. And our statistical analysis showed that there was no difference, significant difference between the fish, the pH system for our conventional and that of the fish that were fed with the canola meal. Secondly, we looked at temperature. Again, we can. This is for tilapia. We kept them in a warm environment, and the temperature regulated between 25 to about 26 degrees. And also, as we can see, there was no significant difference between that of the fish that were fed with the conventional meal and that of the canola meal as well within the period of 3 weeks for the trial. The next also that we look at is dissolved oxygen. Dissolved oxygen plays critical role in fish respiration, root health, and nitrifying bacteria as well. And in our system, we are able to measure our dissolved oxygen in situ in our system without the need for human errors. And we can see that there was no significant difference between the dissolved oxygen between that of the fish meal and that of our canola meal trials as well. Finally, we looked at the electrical conductivity, and electrical conductivity, as we know, it measures the concentration of dissolved salt and nutrient in water, and this indicates the availability of nutrients for plants. And again, on our system, we see that there's no significant difference between electrical conductivity that was measured for both treatment that we had. So I'll take you next onto the results that we had for our fish and plant growth. First is the fish. We started with an initial weight of about 140 grams, and we critically made sure that there was no weight difference between our treatment, that of the canola. If you look at the color coding, the blue, we stand for our conventional fish meal, and the green, that of the canola. Our initial weight was closely about 140 grams. After the trial, the final wet weight was measured and they were about 180. So we see a difference, a percentage weight gain of about 30% between the canola and that of the conventional fish meal. Even though we see some significant difference, there is no statistical difference between the weight gain of that of the fish for that were subjected to the canola meal and that of those that were also subjected to the conventional fish meal. The other growth performance metrics that we also looked at were the specific growth rate, and this tells us the percentage increase in body weight per day, and higher specific growth rate indicates faster growth. So we can see that for our canola, the fishes that were fed with the canola meal had a slightly higher specific growth rate than that of the those that were fed with the conventional meal. However, there was no statistical analysis showed that there was no statistical difference between the two. And the next was the feed conversion ratio. And this feed conversion ratio tells us the amount of feed that is required to produce a unit mass, unit fish biomass. And theoretically, we know that lower FCR or the feed conversion ratios means a better feed efficiency. And as we can see on this graph, our canola meal proved to be a better feed efficiency than that of the conventional fish meal. But statistically, again, there was no statistical difference. But if you look at the graph, there was just a slightly lower feed conversion ratio that was observed for the canola meal. For our plants, like I did mention, we made sure that we started with a uniform seedling that was about the same height and biomass as well. And after our trial, we measured the final wet biomass, and we can see that the growth rate of the fish also increased significantly for that of the canola meal and that of the those that were fed with the conventional meal. But we see a slight increase of that of the canola meal. So which means that the canola meal, the fish that were fed with canola meal provide better, the plant that were fed with canola meal provided better growth rates, had a higher biomass than that of that were fed with the conventional fish meal. And in terms of their dry matter content, we know that the dry matter content also tells us the percentage of their weight remaining after removing all the water, because after harvesting them, we dry them in an oven for a set period amount of time to be able to get all the water out of them and be able to determine their dry matter content. And this is critical for assessing the nutrient quality and overall nutrient accumulation within the system. So for the shoots, we see an increase in the dry matter content for the plants that were fed with the canola meal as compared to that of the plant that were fed with the conventional meal. In terms of their roots, we saw a decrease in the dry matter content of the roots of that of the canola meal compared to that of the conventional fish meal that we observed. Getting to the major analysis that we were most really also very much interested in was the nutrient mass balance. And we wanted to be able to study and know where all this nutrient goes into the system. And like I did mention, in aquaponics system, the major source of nutrient comes from the fish feed that input into the system. So, and just little amount that also comes from the water. So we started with our fish feed, and this graph shows the nutrient input proportions of that of our fish feed and that of our water. The water that we started with was aquaponic water and also city water treated with sodium thiosulfate. And with the exception of copper, with the exception of molybdenum, you can see that there was no nutrient contribution from the water. All the nutrient contribution for molybdenum came from the feed. But for copper we measured a higher amount of copper from the water that was introduced into our system than that of the canola meal. So this tells us that the system, the nutrient input that went into the system has high amount of copper coming in from the water, and that is higher than that of the one that we measured from the feed. So after this nutrient gets into the system, where do they go to? So here is our nutrient output proportion, and as we can see from the color coding, greater amount of the nutrient as expected were lost due to evaporation from the system. However, if you look at the color coding, we see that a greater amount of our nutrient after the evaporation were left in the sludge. They were left in the sludge. Secondly, the next that you can see is in the fish as well. And for instance, when you look at the color coding, you can see that there is a lot of amounts that are left in the fish as well. Then the next that we'll see on the color code, based on the color coding, is on the solution. You can see that some amount of nutrient were also left in the solution as well. And finally, the shoots and the roots, they had very least amount, which as expected. But the overall take home for us here is that indeed we can be able to tell where these nutrients go to and how they help supplement our plant growth and fish growth in our aquaponics system. In summary, we effectively used our Raspberry Pi system to monitor and control these water quality parameters. Like I said, they are very critical in aquaponics system, and they help ensure that the system maintains balance. And this, through the use of the Raspberry Pi systems, we avoided human errors, and also this significantly reduced the labor that takes time to measure all these parameters. And we also saw no significant difference in the water quality between the treatment that were fed with canola meal and fish meal as well. And also no significant difference in the fish and plant production between those two treatments. And we also saw that the nutrient allocation, greater amounts were left in the sludge than to the fish, the solution, and the rest were distributed among the shoots and roots. We can therefore say that the use of canola meal reduces feed costs and dependence on FM, and thereby this improves economic benefit of it. So in future work, after successful inclusion of the 33% of this canola meal into the fish meal, replacing them with the fish meal, we want to increase the canola meal to about 45 and 50, from 40 to 50, and see the impact on the fish growth, the water quality, and the plant growth as well, the plant performance. We also have an ongoing trial with the 20% for rainbow trout in a cold temperature to be able to also determine the impact that will also have on fish growth, the plant health, and overall water quality in an aquaponic system. And depending on the outcome of our ongoing trial, we are either going to increase our canola meal substitution or decrease our canola meal substitution, and which we'll very soon get to know. In future, we also hope to be able to compare nitrogen and phosphorus cycling between treatment that are fed with canola and fish meal as well, because the goal of this system is to be able to advance through a zero waste systems, through the phosphorus discharge, because this is a large major concern in recirculating aquaponic system, and it is the main regulated nutrient in discharge water treatment. So we want to be able to compare these two nutrients and see the impact that it has, and also finally be able to assess the economic benefits to potential adoption of canola meal as a suitable supplement, as a suitable replacement for fish meal in fish feed production and also in aquaculture and aquaponics. With that said, I would like to say thank you, and I would like to thank this, our funding partners, SCAP, Alberta Canola, Sax Oilseeds, Canola Manitoba Growers, Canola Council of Canada, and the Government of Canada. And I also like to thank my colleagues here at the Integrated Agriculture Technology Center for their support. They've been immensely helpful to me because I'm someone coming in from a different background as a chemist. So this is relatively new to me, and I've been learning a lot from them. So I want to say thank you to my colleagues here. And finally to Lethbridge Polytechnic for the space and everything to be able to do this research. Thank you.
So my name is Ivan Tankowski. I'm the Director of Nutrition and Feed Tech at ONDA. And in the next 10 to 15 minutes, I'm going to talk about canola meal performance for Atlantic salmon. This is the project that spans now over a year and a half with multiple sub-projects within it. And I'm going to cover quite a lot of information, so just bear with me. The outline of this presentation will be first short background and research objectives, and afterwards I'm going to talk about each individual project within this bigger project. So the topics will be digestibility of canola meal, growth and general health, pathogen screening in vitro and in vivo, and the last point will be a life cycle assessment of canola meal. As a background, I just want to put some context around what we did with this project. If you think about salmon feed, these are compound feeds that could include anything from 5 to 30 or more ingredients. So when a nutritionist wants to formulate a diet, they look for a specific characteristic of the ingredients that are used. And these ingredients can be broken down into several subcategories that could be proteins, fats, that could be functional ingredients, minerals and vitamins, and fillers. So in this instance, we used canola meal as a protein ingredient. And when a nutritionist looks at protein ingredients, there are 3 main characteristics that they'll be interested in. This will be whether this ingredient is affordable, whether it's available, and whether it has a good nutritional profile. In this table, actually, I don't know if, can you follow my mouse here, or is it just me seeing it?
No, we can see the mouse.
You can see the mouse?
Yep.
Good, great, okay. So I compared here canola meal against common protein sources in salmon diets, which are soy protein concentrate and fish meal. And these are considered sort of the gold standards in salmon nutrition. So what we see here is that even though canola meal comes short on the crude protein, the price is actually much lower compared to these ingredients. So through a careful balancing of the remaining ingredients in a meal, actually canola meal can be incorporated and can be still contributing to the overall protein in a diet in a meaningful way. So in this way, we can answer the question whether the ingredient is affordable. We know that the industry in canola industry in Canada is huge, and there is a readily available canola meal throughout the year. And this is not the case with other ingredients, particularly novel protein products, which are perhaps in short supply or they require significant scale up. The last bit about the nutritional profile we'll see in the next few slides. So the research objectives of this project were to assess the digestibility of canola meal in salmon diets. Then following on this, we will assess the growth, feed efficiency, and general health of salmon. And afterwards, we'll look into the pathogen-specific health benefits, both in vitro and in vivo, and finalizing this project with an environmental impact through life cycle assessment. The first study we did in tank was a digestibility study. The digestibility study is a meaningful way of telling us how much of the whole diet, all ingredients, and then individual ingredients are digested by the animals. So if your input is, let's say, 100% and the output is 20%, you can say that the digestibility is 80%. How do we do this? Well, we start with formulating a reference diet, which is a diet that meets the nutritional requirements of the fish of this particular life stage. And then to test what is the digestibility of the ingredient, we formulate several treatments. So we replace part of this reference diet with canola meal as a whole. So in this case, with this project, we substituted the reference diet with 10, 20, 30, and 40%, meaning that in the last diet, we had 40% canola meal, and the remaining 60% were reference diet. We test each of these ingredients in triplicate, and we stock the fish in 100-liter tanks with modified Guelph system, which allowed us to collect passively the waste of the animals for further analysis. This study lasted 62 days, and we used 50-gram fish as a start weight. This is the formulation we used in this study. Now, the one important bit for digestibility studies is that we used a specific ingredient called indigestible marker. And this is usually an inert metal that is safe for the animals. And it acts like, imagine it like a dye. It goes in the animal and it goes out, and none of it is digested. So through a formula, we know if we analyze how much of this marker is in the feed and how much is in the waste, we can calculate how much of the nutrients have been absorbed and digested by the animals. What did we see in the results for this study? Well, looking at the ingredient level, the canola meal showed improving digestibility in terms of dry matter and energy on the left side of the graph. So we saw that with increasing levels of canola meal, the digestibility also increased. And we saw the same picture with protein and total amino acids. Now, what's interesting here is that our hypothesis was that the digestibility should remain consistent from 10 to 40%, but we actually saw an increase, which tells us that likely there was some sort of interactive effect between the canola meal and the remaining ingredients. And also that the fish may have been forced to use more of these ingredients as there was less of the other ingredients available in the diet. But regardless of that, what we saw here is that the macronutrients were showing good digestibility for canola meal. Moving to the overall digestibility of the diets, we saw somehow an inverse picture for energy, the red dots, and dry matter for the gray dots, as they were dropping in terms of digestibility as the canola meal level was increasing. For protein and total amino acids, we saw that there was a drop from the control, from the reference diet to the treatment diets, but this, in the context of digestibility in general, we are still talking about digestibility between 88 and high 80s and low to mid 90%, which overall indicates a very good digestibility of both protein and total amino acids. So the take-home message from this trial was that canola showed good digestibility, and therefore we proceeded with the next trial, which was our growth or performance study. In this case, we formulated a control diet, again, which meets the nutritional requirements of the fish of this life stage. And we did not include any canola meal in this diet. And then we tested 3 treatments with 5, 10, and 50% canola meal inclusion. The responses we were looking from the fish was general performance as feed intake, growth and nutrient retention. And for general health, we're looking at blood biochemistry, liver and intestine histology. So this study lasted for 103 days, and we used roughly 250 gram fish. So each of these diets was tested in triplicate. In this case, the formulation was more critical because we needed to formulate all diets to have the same overall nutrient composition, meaning that each diet had to have the same amount of protein and lipids and ash alongside other micronutrients to ensure that if we see any differences, they can be attributed entirely to the change of this individual ingredient. So our control diet did not include any canola meal. And then our CM5, 10, and 15 diets included they had 5, 10, and 15% of canola meal. And to include this canola meal, we removed some of the other plant protein ingredients in this formulation. And in this case, this was soy protein concentrate, which is also widely used in Europe, as European formulas do not contain any animal proteins besides fish meal. So what did the results show us? Well, on the left graph, you see the feed input. We did not see any significant differences between the control and the 3 treatments, telling us that first, the feed intake was the same across all diets. And as a consequence, this also means that there was no palatability issue. So even at 15%, the fish was accepting readily the canola meal. In terms of weight gain, we saw the same picture between the control diet and canola meal at 5 and 10%. And at 15%, we saw a marked decrease in weight, significantly different from the control diet, telling us that the fish was not utilizing the feed as the other treatments. And as a result, as Emmanuel already said, the feed conversion ratio is quite important metric that tells us how much feed is needed for an animal to gain 1 kilo of body mass. So in this case, we saw that the control diet, CM5 and CM10, performed the same. So that means that there were no differences between them, while the CM15 showed significant difference from the control and CM5 group. Just to put this into context, fish is arguably the most efficient feed user across all domesticated and farmed animals. And to compare this with other farm animals, for instance, chicken has 1.5 to 1.8 feed conversion ratio. The pigs use between 2.7 and 3.2 kilos of feed to gain 1 kilo, and cows or beef, they go between 6 and 10% and 10 FCR, which tells us that they need up to 10 kilos to gain 1 kilo of body mass. So fish is extremely efficient user of feed, and anything that improves this efficiency is really welcome to the industry, because the biggest cost for any farmer, fish farmer, is feed, and feed can contribute up to 50% of the overall cost on a farm. So in terms of performance, this is the picture that we saw. So the conclusion here is that up to 10% in canola meal supported normal growth and feed conversion. The general health of the animals was also, we saw a favorable picture. We did not see any differences between the control and any of the treatment groups in terms of plasma biochemistry. So the immune status was unchanged, gut health remained unchanged, and liver health was also, there were not notable morphological changes or signs of inflammation. And particularly for the gut health, this is very important because plant proteins often contain something called anti-nutritional factors. And these factors can have severe impact on the overall gut health. And as a consequence, it may have an impact on the health of the animals and can be as a secondary consequence of this, we may see disease outbreaks and mortality accompanied also by poor growth. So not seeing significant changes in the gut inflammation score was quite important in this particular case. So that was the second trial we did. Then we moved to the pathogen screening, first on the bench or in vitro. We screened 9 known pathogens that affect Atlantic salmon. And what we looked for here is if there was any inhibition of growth of these pathogens using extract from canola meal. So from the 9 screened pathogens, we saw that there was an inhibition of growth for 2 of them. One of them is called Tenacibacillum finmarkensi, and the other one is Streptococcus ine. The results for ine was not as pronounced as the one for Tenacibacillum. So our focus stayed with Tenacibacillum. There are several subspecies within this group that affect salmon. And this particular finmarkensi is the one that affects the salmon industry in Norway. While the ones that affects the industry in Canada is called Tenacibacillum maritimum. So we saw that there was about 20% reduction of growth in this particular finmarkensi case. And we wanted to investigate whether these on the bench results will translate into real life results in vivo. So we designed a study based on the results from the in vitro study where we challenged the fish with each of the 2 Tenacibacillum species separately and then in a combination as a mix. And the fish was split in 2 groups. They were fed either a control diet without canola meal or the second group was fed canola meal 15% containing diets. So this study lasted 106 days. We fed the fish for 5 weeks of the treatment diets, and then they were challenged. What we saw as a result is that in the case of Tenacibacillum finmarkensi, we saw the same results as we saw on the bench. So the results on the bench were confirmed by this study as we saw 53% improved survival when we compare the control group versus the canola meal 15 group. And this was, as I said, important verification of the results we saw on the bench. When I forgot to say that this actually Tenacibacillum finmarkensi causes a disease which is called mouth rot, and it may result in severe mortality events on the farm and downgrade of salmon because they have ulcers all across their body. It starts with the mouth and yeah.
Sorry to interrupt. I just want to let you know we're at 15 minutes. So if you could try and wrap up in a minute or 2, that would be great. Thank you.
Yeah, absolutely. Okay. So the take-home message here that we saw improved survival against finmarkensi. The last part of the project included the life cycle assessment, and the question we asked here was if we added 10% canola meal, would this reduce the environmental impact of salmon production in Atlantic Canada? The impacts that we assessed were global warming potential in terms of kilograms of carbon dioxide equivalents, acidification, fossil energy use, land use, and water use. The output of this study was that if we added 10% canola meal to diets for Atlantic salmon used in Canada, we saw a decrease in carbon dioxide equivalent emissions by 10% in fish grown in sea cages and 2% in fish grown on land. The water use dropped by 21% in sea cage and 6% for salmon grown on land, and the land use dropped by 3% in both groups. We saw a minimal increase in acidification, while the fossil energy use remained unchanged. So the conclusion of this whole project was that canola meal can be included in Atlantic salmon diets. It has a good protein and amino acid digestibility. It showed comparable performance between the control and the treatment groups and supports normal health. It may offer a mechanism of protection against Tenacibacillum finmarkensi, and it reduces the carbon dioxide equivalent emissions of salmon that are grown in Canada. These are the acknowledgements.
This was originally planned to be presented by my colleague Shawki Ben Shah, but he's away. And this is actually a collaboration between 3 different teams of Agriculture and Agri-Food Canada. Shawki is in Sherbrooke, Stephanie is in Lethbridge, and myself is in Quebec City. So actually, first a small introduction about greenhouse gas emissions. So it's about, I mean, everybody are going for reducing greenhouse gas emissions and for the goal of reaching net zero emissions by 2050, because methane is almost 16% of the global greenhouse gas emissions, and more than a quarter of this methane is coming from enteric fermentation from ruminants. So it's important to have effective mitigation strategies to reduce enteric methane emissions from ruminants. So if we look here at the greenhouse gas emissions from the dairy production cycle, we can see that enteric methane emissions are almost half of the emissions from dairy production. So because we want to reduce these emissions, it's going to be advantageous to target these emissions and to mitigate methane emissions from the enteric fermentation. Seaweed has been suggested as the mitigation strategy for methane emissions. So we have 2 sources of seaweed that is effective against methane: the red algae and the brown algae, and they reduce methane emissions by almost by 65%. But because this species of red algae that is specific for reducing methane emissions is not native to Canada, and importing these species into Canada might create the biological problems because it's not a native species in the northern hemisphere, and it might the shipping and the packing might increase greenhouse emissions. And it has shown that it has adverse effect on intake and milk production. So the alternative was to cultivate seaweed in a controlled environment and to characterize their nutritive value and to monitor their secondary metabolites that have an effect on methane. So actually, why did we choose to do this work? First, we had a work published in 2021 with Shawki, myself, and other members of the team, and we saw that increasing the incorporation of canola meal into the diet of dairy cows would reduce methane emissions by 6%. And when we express this reduction based on milk produced, it's about 10%. Also, in an unpublished study, we saw that incorporating seaweed in the diet of dairy cows would reduce methane emissions by almost half when the emissions are expressed on milk production basis. So the proposed work combining these 2 strategies and see their effect on enteric methane emissions. So the objective of the study is to assess the effect of using a cultivated dried seaweed that will be referred throughout the presentation as E in the diet of the lactating dairy cows fed canola meal or soybean meal. So we're looking at variables of dry matter intake, digestibility, milk production, nitrogen excretion and use efficiency, body weight, rumen fermentation, and protozoa population. We're also looking at antimethane emissions in the respiratory chambers, and we also looked at the methane emissions from manure. Our colleague René Petry is also looking at the microbiome and the effect of these treatments on rumen microbial population. And at the end, we will make a life cycle assessment of these anthropic methane mitigation strategies. So for the purpose of this project, we used 16 cows. 12 of them were ruminal cannulated, and they were almost in their third lactation. And they were in 115 days in milk, and they weighed about 730 kilograms. So we used a 4x4 Latin square design of the experiment. So that means that each cow will receive the treatment, and we will compare the cows between each other, and the cow will receive this treatment within 35 days. That includes 14 days of adaptation and 21 days of measurement of the variables. So the treatments were soybean meal with seaweed, canola meal with no seaweed, and canola meal with seaweed. And the feeding percentage of seaweed was 0.18% of dry matter intake of dry matter intake. So to provide the cows with seaweed, we mixed it with 50 grams of protein supplements, and then 1/2 kilo of the total mixed ration. And then we fed this to the cow before feeding the whole diet to the cow. And we did that twice daily. And we milked the cows twice daily in their stalls. So here you can have an outlook on the chemical composition of soybean meal and canola meal. And this is the classical differences between the two meals, as previously my colleague who used them for feeding salmons showed that there's a difference in crude protein content, but also there's a difference in their profile. But according to National Research Council and NASEM, the RDP and the RUP of the two meals is not different. And because there is more fiber and more lignification of the fiber in the canola meal, the concentration of energy in canola meal is a little bit smaller than that of soybean meal. For the other part of the treatment, the seaweed is a mixture of cultivated of red seaweed and brown seaweed. And this is a mixture that has been prepared by the company Synergrace in Alberta. So we're looking here at the two diets that were offered to the animals. So we can see that the diet was composed mainly of corn silage, grass and legume silage, and hay as forage sources. And the ingredients of the diet were similar between the two diets, except for canola meal and soybean meal. To replace soybean meal with canola meal at equal basis, we also included soybean hulls in the soybean meal diet. So we can obtain an inclusion rate of 24.5 canola meal in the diet of dairy cows. And we can see that canola meal is the only source of protein supplement in the diet of these cows. So here this is the chemical composition of the diet, and we can see that the two diets contain the same level of crude protein, and they had the same level of net energy of lactation, and they almost had the same level of rumen degradable protein and rumen undegradable protein. So here you can see about the facilities of methane measurement that exist in Sherbrooke. These are the respiratory chambers, which we put the cows in these chambers for 2 days, and we measure their intake. We milk the cows in these chambers, and because the cows have windows, the chambers has windows, so the cows can see through these windows the other herd, and they can see the sound. They can hear the sound of the other cows, so they're not threatening the chambers. So they maintain their intake and milk production as if they were outside the chambers. So the variables that were measured were analyzed statistically using a Latin square design with a factorial arrangement of treatment, with a protein source, the seaweed, and their interaction as fixed effects. Well, and the random effect was square and cow within square, and we signified that there's an effect of treatment on the variable when the p-value was less than 0.05. So now for the results. As you can see, the molecule that is active in reducing methane production in the rumen of the cow is the bromoform, and the bromoform, as you can see here, it's the carbon attached to 3 atoms of bromide and 1 unit of hydrogen. And as you can see, throughout 2 months, the concentration of the bromoform in the product that was produced by Senegraze was stable throughout a period of 2 months. So the concentration was about 4.65 and varied by 0.225 milligram per kilogram dry matter. This is a previous experiment that showed the effect of methane emissions of adding seaweed or the sea to the diet of the cow. As you can see here, in the blue is the methane production throughout the day of a cow that did not receive bromoform. However, you can see in red when the cow receives seaweed that the methane production really is inhibited and reach a low level of about 2 grams per hour compared to 30 grams per hour at the lowest and the highest point for the differences between the seaweed supplementation and no seaweed supplementation. So looking at the results of our experiment, we saw that the dry matter intake was not affected by the meal. So it was similar between soybean meal and canola meal, but adding seaweed reduced intake in both treatments, and it reduced dry matter intake by 1 kilogram in soybean meal and by 2 kilograms in canola meal. When we looked at milk production, we saw that when cows receiving soybean meal were supplemented with the seaweed, there was no effect on milk production, but there was a decline in milk production when the cows were fed canola meal and supplemented with seaweed. Still, we can see the results that we have seen in a previous project, that when compared control soybean meal with control canola meal, cows fed canola meal produced higher milk production than those fed soybean meal. Looking at yield of fat and protein, we can see the same effect, that there was no effect of supplementing seaweed to soybean meal on fat yield and protein yield, but there was a decline in fat yield and protein yield when seaweed was supplemented to cows fed canola meal. The nitrogen use efficiency averaged 30%, and it was increased by seaweed supplementation due to a decline in dry matter intake and nitrogen intake. So here's yes.
Just wanted to let you know we're approaching on 15 minutes.
Okay.
So if you might be able to wrap up in a minute or two, that would be great. Thank you.
Okay, awesome. So just going through this fast, we're looking at the rumen fermentation. We can see that supplementing seaweed shifted the rumen fermentation toward less acetate and more propionate. And the main results showed that canola meal still reduce methane production when expressed on gram per day or gram per kilogram dry matter intake or on milk production basis. And supplementing seaweed also reduces the production of methane almost by half. So when we combine the two strategies together, we can see that canola meal and reduce anthropic methane production by 9%. When adding seaweed, the reduction is 33% across the two treatments. But when we compare canola meal plus seaweed versus a classical diet made with soybean meal without seaweed, we have an added reduction of 39% of greenhouse gas emissions coming from anthropic methane emissions. These are the results of the nitrogen intake. They're not complete, but the team is working hard to complete them. But we see that there's a decline in urinary N excretion due to a decline in N intake. So in summary, canola meal versus soybean meal, there is no effect on dry matter intake. There is a small effect on milk performance, but there is a decrease in anthropic methane emissions. Feeding the cows, the red seaweed reduced dry matter intake by 6% and reduce milk production in cows fed canola meal. And it reduces anthropic methane emissions by 33%. But as I said, when combining the two strategies together, there's a larger decrease in anthropic methane emissions. For further work, we want to look at long-term adaptation of seaweed in cows. We would like to see a dose response effect so we can measure the optimal seaweed feeding rate for cows fed different diets, and that would lead to the question of seaweed and diet interaction. We're in progress of analyzing seaweed residues in milk and see the effect of seaweed on methane emissions from manure. So I like to thank the research team and the financial support of Agriculture Canada and Canola Council of Canada. Thank you.
I'm happy to talk about canola meal. I've been involved with some projects with the Canola Council now for quite a while, I think almost 15 years. So it's been a while. And I'm happy to talk about our current project, which is the impact of canola meal for sustainable dairy cow product or dairy cow diets. And the student working with me is Alex Coops, and I'm working in collaboration with Dr. Sebastian Areola and Dr. Laura Hernandez, both at the University of Wisconsin. A lot of people have given some background already on why canola meal is a great fit for livestock diets. In dairy cattle, there's a growing body of work that really shows a great match of feeding canola meal in high-producing lactating dairy cow diets, and a lot of it's related to amino acid profile, but also we believe energy is also a great part of it, and the fiber that is in canola meal helps in the rumen and ruminal fermentation. This graph really just kind of depicts how canola meal matches up with other protein supplements, and the bottom being rumen microbial protein, which is really what we're trying to match the best with. So canola meal, other than fish meal, which is an animal protein and we don't really use anymore, is probably one of the best matches to ruminal microbial protein. And when we feed it to cattle, it makes a great match in both energy and amino acid profile that's needed for the cow. So it's a great match in that respect. We've done actually 2 studies here in early lactation. When I first came here about 10 years ago, we did a study comparing canola meal and soybean meal at 2 different protein levels. And in this particular experiment, we actually had more of a high corn silage diet. So the graph depicts that the corn silage was almost 40% of the diet, whereas the alfalfa was about 15.4. So we were feeding about 55% forage in the diet. And in this particular study, it was the first one where we were feeding early lactation, randomized complete block from the start of calving through 16 weeks into study. So 4 months into the lactation. And the cows averaged over 4 kilograms more milk per cow over that entire 4-month period, which we found quite astounding at the time. A lot of the literature in mid-lactation shows, 1.5 kilogram increases, but nothing as dramatic as 4 or greater. And we attributed that difference to a better amino acid profile, but also likely better fermentation in the rumen. So we did follow that up with a second experiment in the last canola cluster, and that we were interested in comparing canola and soybean meal with diets that were more equal in forages. So we had an equal 50%, 50-50 with corn silage and alfalfa, and so about half of it. And these diets were formulated at 60% forage, so a little bit higher, but 30% coming from corn silage and 30% from alfalfa. And we also looked at the dry period on whether or not feeding canola meal during the dry period was valuable in carryover into lactation. The results from this actually showed there wasn't really carryover from the dry period. However, feeding canola meal during early lactation still had a big impact, and it was almost 2 kilograms per day. But it wasn't quite as dramatic as the first study, which was 4 kilograms per day, which led us to the current project, which is, are there interactions of the types of forages we feed and the fiber digestibility that occurs in these forages interacting with the protein supplements that we feed, namely canola meal versus soybean meal, which led us to our current study where we wanted to further investigate how different forages in combination with canola meal resulted. And so that leads us to our current study, and I'm going to have Alex explain where we're at. We just started actually in January, so we're kind of at early stages. We don't have actual results to present, but we can give you an update on where we're at and what we're all doing. So with that, Alex, hopefully you're in the middle.
Can you see me all right?
Yes, we can.
Perfect. So thanks, Ken. So I'm going to discuss the current research that we're doing now here in just outside of Madison at the USDA farm here. So currently this research aims to investigate the utilization of canola meal in the early lactation dairy cow with corn and alfalfa silage as the forage sources. The objective is really to build on the previous 2 studies that were done here to better understand the efficient and sustainable conversion of canola meal into milk. So in the study here, we have 4 treatment diets. These diets were formulated as isonitrogenous diets with canola or soybean meal as the main protein source, and with corn silage and alfalfa silage in 2 combinations in a 2x2 factorial arrangement. The experiment was designed with 80 multifarious early lactation Holstein dairy cows with 20 cows per treatment in a randomized complete block design. These cows were enrolled in calving. Prior to calving, they were fed a 2-week common dry cow diet. And at calving, they were then assigned their treatment diets, and we followed them for 16 weeks. We are following them for 16 weeks. So a little bit more into the diets. Like I said, we have 2 protein sources. These are the main protein sources in the diets of both canola and soybean meal. And then we have 2 different forage combinations here with diets A and C containing 40 and 16% corn silage and alfalfa silage on a dry matter basis. And diets B and C had equal amounts of corn and alfalfa silage at 28% on a dry matter basis. So we, for data collection here, we're collecting an array of production data, which includes milk yield, milk composition, and dry matter intake, to sort of help with this production data. We collected methane production data and digestibility to sort of assess animal efficiency. Then on a molecular level, we're collecting blood samples and tissue samples, which include liver, mammary, adipose, and muscle samples. And when we take all these samples together, it sort of helps to paint a real comprehensive picture on animal performance, not only on an applied level, but also on a biological and physiological mechanistic side of things with relation to how canola meal interacts with forage source. So currently, we've had 60 cows that have calved, with 20 left to calve to make up our 80. We've had 16 cows that have unenrolled from the study, so they're already done, and we've completed all our biopsies of the select cows that we wanted to biopsy. And currently we are still sampling at the farm every week, and we're sort of ramping up to do some analysis in the lab. And so the real outcomes that we're hoping for here, or the focus of this research, is to better understand production responses to canola meal supplementation and early lactation. Also to help understand the mechanisms of canola meal supplementation through biopsies, to investigate the effect of protein source and forage source on amino acid utilization, and finally to determine methane production from protein and forage source supplementation.
We'd like to acknowledge our funders, the Canola Council of Canada, and the partners involved with that, and also with USDA and the University of Wisconsin Dairy Science Department, at Dairy Animal and Dairy Science Department. Just as a follow-up, this is the first study. We have a second study that will be planned further into the project, and hopefully data that's generated will be part of a LCA analysis that we are planning along with others involved with the dairy project. So that's a summary of where we're at so far. So I'll be happy to take questions during the Q&A session. So thank you.
Okay, well, I'll hit everybody with one question. We'll see how much time we have left. First off, I'd like to thank everybody. Just fantastic job. I think it's really exciting research, and we're really moving ahead. I'll start with Emmanuel. I know you were just feeding these fish for a very short period of time, but I think we've got some curiosity from the audience. They'd like to know if you feel that the type of diet that these tilapia are being fed have any impact on tissue quality ultimately.
Right. So far, we haven't seen any difference in the tissue quality between the type of diet that we have, but I think it is something that maybe in the near future we will look deeper into that to see if the type of that could play some significant role in the type of tissue that both diets could produce in the fish. It's something we could consider. Yeah, that's a very good question.
Okay, Yvonne, I think there are some people hoping that we can use canola to, really if we were to get that incorporated in diets, what do you feel the market size would be for canola meal?
Well, depends on, can you hear me actually? Let's start with that.
Yes.
Yeah, okay. Depends on the market. Canada has a problem with the salmon growth at the moment. Actually, we're seeing the opposite due to the federal decision to close the farms on the west coast. So we are seeing for the past few years decrease in the production volumes. This may change, of course, and as far as I understand, the East Coast is also looking for to increase their output. But currently, I think the production stands around 60,000 tons annually. So if we consider the feed conversion ratio of 1, then we are talking about maybe 6,000 tons of canola meal within Canada. But the study we did, we also benchmark canola against soy protein concentrate, and then we're talking about the European market, which is huge. Norway alone is, I think, over 500,000 metric tons a year. So there's a huge potential for canola if it gets adopted by feed producers and farmers to hit much higher volumes.
Thank you. Fadi, I noticed that there seemed to be a considerable difference in the response to seaweed, and almost a negative effect with canola meal, but not so with soybean meal. And why do you feel that would be the case?
Well, actually, there seems to be some different studies showed that there's an interaction between diet and seaweed supplementation. So that's probably that probably needs to be investigated more. Luckily, in our team, we have Renee Petrie. She's doing rumen microbiome. So with the analysis of the rumen microbiome, we can see which microbial populations got diminished or flourished between the two diets that were supplemented with seaweed, and it might lead to more understanding with that. Probably because I would think that mostly the decline was due to a decline in dry matter intake, and the decline in dry matter intake was probably more because of the seaweed with the canola meal initiated more propionate, which has interference with the satiety of the animal. So probably with the canola meal, we need to look at further research on reducing seaweed so we can have the same level of the reduction in methane emissions, but at the same time, no effect on milk production.
Okay, thank you very much. Kenneth and Alex, I know that your base studies are based on forage, being from the Forage Institute, and I think it's probably important, and we're not looking at it enough. And do you feel that the type of forage or the quality of the forage would have an impact on some of these studies?
Great question. I think the answer is yes. And it's the premise of what we're doing. So both the type as well as forage quality is the premise of the two studies we're proposing. And so the one we're currently doing is really a follow-up to what the literature suggests right now, which is higher corn silage diets often see a better or a greater response in the addition of canola meal compared to alfalfa-based diets. And I'm not really certain why that necessarily is the case, except probably with the nutrients that's being provided. We do balance for like starch across the board and the macronutrients. So what is it about corn silage other than there's potentially more energy in corn silage? And so the current study was really a follow-up kind of our last two, where we saw more dramatic response with the high corn silage diets. And others have started seeing that as well, where with corn silage, they see a better response. As a follow-up to the quality question, I do think the quality of the digestibility of the forages also will have an impact, and that's really what the second study is going to be on, and looking at how the quality of the fiber that's in the forages that we're feeding can impact in combination with canola meal. So I'm really looking forward to that study. We're still working on how that's going to be done, because formulating diets is really part science and part art, as you know, Essie. So we're in the process of working on that right now, and having the forages that we need for the next study. So we're going to be planting things probably for this coming year. So that's kind of my summary. I don't know if you have anything to add. Okay, that's it.
Okay, thank you.
We might have time for one more quick question if there is one.
Well, we'll pick on Emmanuel.
Okay.
I thought it was interesting that the root development, you had less root and more plant when it came to the lettuce samples. Is that a good development? Like, it would seem like you've got more of the edible component and less of the part that might become waste.
Sorry, please, the question again. I kind of missed the first part.
When you had only one feature that seemed different in your trial, and that was the composition of the lettuce, and you had more of the plant component and less of the root. And that to me seems like it should be a good thing, should it not? And
yes. Yes, if I understand your question correctly, you are talking about the nutrient output proportion that I showed, where we saw greater amounts of the nutrient going to the shoot as compared to the root. Yes, it is because it is based on what we saw from the dry matter content. We saw a higher amount of dry matter content in the shoot than that of the root. So that correlated that high amount of nutrient went into the shoot than that of the roots.
Okay, great. Thank you. Thank you, Haley.
Thank you very much. It's a pleasure to be here. I'm an agricultural economist working with Simon Wassine. That's also an agricultural economist working with me. We're responsible for doing the economic impact assessment as part of the Canola AgriScience clusters. Today, what I want to talk about is a little bit of background to where this came from, the rationale, the requirements for the assessment, some of our timelines, and then seek some of your input on as we go forward. As you know, this is our fourth AgriScience cluster, if you like, starting with Growing Forward 2, CAP, and now we're in SCAP. It's part of the last SCAP cluster. There was a requirement for all returning clusters to do an economic impact assessment of the previous cluster. And for the canola group, that's the CAP cluster is the previous cluster. But as I'll mention later, some of the individual benefit cost studies can go back further to projects that were in the CF Growing Forward 1 or 2. This requirement was introduced as part of the sort of evidence-based decision making at the federal government, something the Treasury Board has used for a long time. These types of studies are needed to demonstrate economic value, not only to the government, but also to producers, downstream value chain, the Canadian economy. And I think it's a good story. Agricultural research certainly contributes to economic growth, and I suspect that it'll be we'll have some good news at the end of this process. It seemed to be locked up here just a sec. Here we go. There's two components. There's a qualitative and a quantitative component. The qualitative requirements are basically designed to look at how effective the overall effectiveness of the previous cluster. So it has to be the one previous to SCAP. It looks at the overall cluster, looks at individual themes, looks at how the research is coordinated, the synergies, economies of scale, looks at sector level impacts. It also looks at each of the activities, it reviews the performance report, develops a scorecard for each of those activities, and it evaluates it based on 10 criteria, including personnel involved, training, transfer events, new technologies, scientific impact, etc. There'll be a discussion of commercialization and adoption of the activities, description of the environmental and social and non-market impacts of the research, and it'll also identify areas of success and areas for improvement as part of this qualitative assessment. The second part of the assessment is the quantitative research, and that is a requirement. Oh, sorry. One example of a qualitative impact is the scientific impact, and we've started in on that already. We're using two approaches. One, first of all, is very comprehensive, where we basically do a census of all the research of the team during and post cluster, and that's and by looking at the number of publications, citations, the impact factor of the journals, we get a signal of the overall quality of the scientific team that's involved in the cluster, and I can tell you that it's an pretty impressive set of publications that the team puts out over time. And the second approach is to be very conservative in our screening and look at those activities, those publications that came directly from the activities. It's smaller in scope, but those activities are easily attributed to the research activities that were undertaken by the cluster. For example, if you look at just the publications that were published during the CAP cycle, there was 15 publications, 211 citations with 14.1 citations per paper. It has a Herfindahl H index of 8, meaning that over half the publications had 8 or more citations. These reflect a relatively strong performance of citations and a positive degree of impact on the scientific community. So that's one example of a qualitative impact. It's hard to put any dollars on that, but certainly you could talk about it having a scientific impact. The quantitative assessment requires a benefit cost analysis of 3 activities. A benefit cost analysis looks at the value or the benefits created from the research outcomes from a research activity and compares it to the cost of the research activity that's undertaken, normally expressed as a benefit cost ratio. And if it's a good investment, you want benefit cost ratios greater than 1, and a lot of scientific research typically has benefit cost ratios higher than that. For this exercise, the instructions were to select 3 activities that were likely to produce benefits, have created knowledge with an expected value, and have evidence of a viable path towards commercializations. And when complete, these benefit cost analysis will also serve as success stories for stakeholder and communication and engagement. Where are we at in this process? We've completed literature review, we've developed methodology for the different parts. We're in progress right now in terms of our preliminary qualitative assessment, scientific impacts. We're in the process of selecting the 3 activities for the benefit cost ratio. When that's all done, we will be able to submit a final proposal for the economic assessment, which defines exactly what we're going to do and how we're going to do it. And then over the next year and a half or so, we will complete the qualitative and quantitative assessment and provide the success stories. Just a reminder, the CAP cluster had 7 themes. It had environmental performance in food processing, livestock production, feeding canola meal. That was both swine and dairy. Agronomic studies looking at yield and quality and harvest, etc. Sustainability, climate change, looking at nitrogen use efficiency. Theme 5 had dealt with integrated pest management, and theme 7 dealt with blackleg and verticillium. In order to, there was 33 activities within the cluster, and just as an exercise to start filtering them, I decided to score them using 3 criteria. We need to have knowledge that can easily be attributable to the CAP research. In other words, a research result that clearly came from the activity. That research result has to have quantifiable, large anticipated benefits. And finally, there has to be some evidence of commercialization, on a scale of 1 with 3 being a very probable commercialization of the knowledge that's being generated. I'll share these slides. You'll have a copy of these slides. I developed a list of 7, if you like, out of the 33 with a bit of screening. Each of these activities had knowledge that clearly came from the activity, so they met the so they can be clearly attributed to the activity. The benefits can be quantified to some extent, depending on the size of the market and the type of effect we're talking about. And the commercialization, I've got question marks beside most of those. Sorry, because I'm not aware of it. Haven't started the research to look at that. But these are the types of things we need to do to try to pick the best 3 activities. Also, I've only so far looked at CAP activities, but ideally we'd want to include any of the activities in growing forward 1 or 2 in our list of 3 benefit cost studies. And some of the earlier research is likely to have benefits that are more apparent because it's been they happened a while ago. So I need your input. If I missed any activities in the CAP, but are there other ones that you think would be really important to include? In other words, are there large, successful previous cluster activities with commercialized benefits? And I see some of these themes keep repeating themselves, so I suspect on the disease front, there may be some of those. And finally, once we do a benefit cost analysis, we'll get in very carefully looking at the evidence of commercialization, but it would be at least good to have a discussion and some indication that the knowledge has on the path to being commercialized or is already in the field. If you have some input that would be useful in any of these regards, please email me. Each of you probably know your research or related research pretty well, so you're the right group to ask. If you could email me, that would be useful with any suggestions, and I'll be following up with Curtis, so that will be useful as well to finalize our list of 3. So with that, I'd just thank the sponsors and turn it over back to the moderator. And I guess while I'm waiting for the question, I would also add that all the clusters are doing this, and I'm involved in a few of them. And so the methodology is similar, but it varies a little bit. And Agriculture Canada was was fairly prescriptive, but there's a lot of degrees of freedom within those prescription as well as to how to do this. So we've done a lot of work sort of developing the methodology.
Okay, welcome again to the panelists. Thanks, Haley, for introducing everybody. I'll be the moderator. I'm, as most of you know me, I'm Curtis Rempel. I'm with the Canola Council of Canada. I'm Vice President for Crop Production Innovation. It looks like we have potentially Doug substituting in for Cody. Is that correct?
That's right.
Yes. Okay.
I'm not sure if my camera's working.
Yeah, perfect. You are. So I guess you'll put on both a research administrator and a farmer hat today and give us some perspective. Okay, well, thanks everybody to joining. I have a couple,
we put together a couple of questions, and just by way of introduction as well, we have an innovation committee at the Canola Council of Canada, has a number of different roles, including vetting new research opportunities, evaluating and valuation of research that Richard talked about, and a whole host of other activities that guide the innovation suite here at the Canola Council of Canada on behalf of our value chain. A number of the members are on the panel today are part of that innovation committee or a periphery to the innovation committee as well. So I've sort of tapped all my colleagues for this exercise. In terms of the questions that I'm going to pose, Chris, do you have can you share those slides online with everybody, or does that wipe everybody out?
I certainly can.
Okay, that might be useful. I'll get the first question going. First question is, again, it's something that is very much top of mind for all of our stakeholders in our value chain. How do we balance investments for short and long-term research? And can we adequately address short-term needs as well as longer-term goals and aspirations? So I'm gonna assign different people first of all to get the discussion going, and then hope my colleagues can jump in. But I'm gonna let Wilf Keller kick this question off. He chairs our Innovation Council and is a co-chair of our Innovation Committee, and also chairs the Science Advisory Committee for our Science Cluster. I know, Wilf, you've thought about this a lot, so I'll get your thoughts initially, and then we'll move across.
Okay, thank you very much. I'm sorry I missed a lot of the other sessions, so there may be things that I've said that I should have been including in these comments. However, I think it's important when we talk about investment in research, we need to consider the full innovation pipeline, right from the front end where we generate knowledge and technology through to development, evaluating prototypes, and ultimately commercializing new products, varieties, etc. And I think that pipeline is not fully balanced in Canada at this point in time in terms of agricultural science. We're weak, in my opinion, weaker on the front knowledge generation end. I realize that it's often said that Canada's strong in basic research and knowledge generation, and we need to emphasize commercialization. That's only partly true. We are not as strong as some people think we are in knowledge generation. We need to define some key areas, some key target areas particularly for canola in this discussion, and we need to put long-term timelines on those. You could talk about managing carbon. You can talk about things like the rhizosphere and the microbiome as long-term visionary things, moonshots, if you will. Those need to be identified and funded. So I think it's possible to do this, but it needs a rebalance. We need to put more emphasis on some of the front end with more public, maybe pure public funded research in long timelines to get the ball moving in these critical areas. In terms of balance, that's up for discussion. It could be 20% of our investment into those longer term, more publicly funded initiatives. And the sharing of that is important, with the public maybe having the bulk of the investment at the knowledge end. The producers and others start investing more heavily, and ultimately industry as we move down that pipeline to commercialization. So I would like to just comment about a couple of things here, 3 things to say. We need to also, number one, we need to revisit the view that canola research or canola as a crop is quite mature. It doesn't need as much public investment. That has been an issue. I feel that has been an error, if I can say that needs to be revisited. I could talk a lot more about that. Number 2, I think we need to expand our ecosystem to include more emphasis on startup companies and new companies that can be working in the canola space. These are brilliant entrepreneurs that start these companies. They have excellent tools, but they may not always have the right target. We can help them with that. And then secondly, when it comes to investment, we need a bigger investment pie. we need to look for new players. I mean, for example, Farm Credit has now come up with a $2 billion investment. What's Canola's share of that? What about private investors to help companies move forward? Well, I think there's a lot more resources. I feel very positive that we can have that balance, and that's asked in the second part of this question. Can we actually accomplish the short and the long term? Most definitely we can.
Thank you, Wilf. I'm gonna put Dave on the spot now. For this one, I'm gonna talk to our industry partners first, and then I'm gonna go to our farmer and researchers. So Dave, and then Chris. You're muted.
Sorry, you'd like me to speak to the same question as Wolf did?
If that's okay. Anything
Sure,
to add?
sure.
Yeah.
So I think Wolf framed it up and covered a lot of ground there. For me, I'm a big picture person, and I think, question would be, do we have a set of, and it's going to be a limited number of consensus priorities in the industry? And I know, the grower groups, having sat through, some of their presentations, I know each provincial group has their consensus priorities. Now, these are things that are going to be most important to the grower or downstream. And so, as Wolf said, there's definitely a need to have a clear, sustained investment in what I'll call basic research. That's what was called today. And, that can be fleshed out more. But so I think we need that. And in addition to consensus priorities, and you can start at grower level and then work back upstream. So you accumulate those, and then you go through the messy process of trying to prioritize them. I think we also need to look at the research investment in the industry holistically, and we need to recognize the strengths and the limitations of private companies and what their greatest priority is for the industry versus the various public research groups and the capacity we have there. Sustained investment is critical, I would say, across the industry. Wilf said it should grow. Of course, we always want it to grow, but you turn that around and say, well, can we use what we have today more effectively? So that's where my focus is in terms of saying, have we got the priorities well defined? And is there more or less consensus on those with all the necessary input? And are those visible and constantly displayed? And that's going to be a process always in motion, because those will get updated over time. And then secondly, are we looking at all the capabilities, resources, harnessing all the horsepower of the industry as a whole? We tend to put a lot of emphasis, and I would say load, demands on public sector research, and their role is critical, but I think, we should cast the net a little more broadly, and then, we can see what they do and what they can do most effectively, and we need them to do in light of total industry needs. But then also let's look at, all the other partners.
Thanks. Okay, in the interest of time, Chris, I'm going to tee you up for the next question. I'm going to just talk to one of the researchers here, Sally. You're actively involved in delivering research, and you live it and breathe it every day. But if you think about long-term research versus short-term, what do you think in sort of as you reflect on it, where would a balance lie? If you were thinking about it from a researcher talking to the canola value chain and farmers.
Yeah, and I think my answer will tie in nicely to the stage that that Dave and Wilf set in that really taking a step back and looking at the bigger picture, who can do what effectively, where is the longer term investment required, and is that a better fit with the public industry? And I'm 15 years into my position with canola and with Agriculture and Agri-Food Canada, and something I've really seen a transition from is kind of going after or the opportunity to go after, say, easy targets. So from a breeding perspective, single major genes, versus moving into the more complicated targets, the quantitative traits, the traits where there's lots of environmental impact, the traits that are trickier to phenotype, the traits that require more of a multidisciplinary team to look at. And and I've seen that transition. I think it is going in the right direction within my organization, within Ag Canada. It is recognized that I believe it's recognized that with canola, some of those trickier traits is where the work needs to move towards. That being said, there's still a need to support the industry, with something that's a little closer to market, or something that, say, a higher heritability trait. We still want to be in a position where we can support the industry if there's new players, smaller players, or if, for whatever reason, one of the bigger players is looking for some different innovation, different ideas. So I guess the key is to be flexible so that we can respond to, be in a position to respond to both.
Thank you. Okay, in the interest of time, I'm going to move on to the second question. Those were some excellent thoughts. And this one again links industry and growers. And so are there areas that industry and growers have strong alignment for research investment, and what approaches can help strengthen this relationship when it comes to investing in research? I'm going to turn to Doug, first of all, as sort of the grower research manager for a provincial grower organization, to provide us with some initial thinking. And then Chris, I'm going to turn to you, and then others.
Sure, I'll just kick it off just saying that, so this is a mix of some of my ideas as well as Cody Niggy's, but things where industry and growers have strong alignment would be things like disease resistance trait discovery. So that's more on the medium to long term for investment scale. So one of the advantages of the crop commissions in investing in genetic trait discovery is that they can leverage federal and provincial government spending, which the commercial breeders and seed companies can't access. So that allows government spending to be injected into the equation. And I guess if we left it all for the commercial seed companies to do all of this work, potentially you have a lot of competition and duplication of effort. So each company is trying to find the same set of genes and monopolize access to those. So that leaves less money for each of the seed companies to put into other breeding costs. So that causes seed costs to go up for the farmer as well. And as well, because of IP, no one breeder will have access to all of the best traits.
Thanks, Doug. Any Chris Anderson, thoughts on alignment and how can we strengthen relationships?
Yeah, so I would say, building on what Doug commented on, there's a lot of alignment, I think, between industry and growers and the value chain about what's important. We want to grow more canola. We want to grow it more profitably. We want to know how to use it. We want to help develop markets and trade. Those are all things where we have alignment on. And so whether it's diseases, whether it's carbon sequestration, whether it's hairy canola, loved seeing the update on that earlier today. But it's a matter of, I don't think there's a disagreement about what's important. Sometimes there is arguably a disagreement about the commercialization aspect of that, and when and how a particular discovery should end up in a farmer's hands and what the price would be, as Doug alluded to. And at the end of the day, there is a distinct difference between the research component and the development of a product that can actually be used on a producer's farm. And those processes, I think, if we're going to be efficient, need to be complementary rather than competitive. And Doug, I think you alluded to that. Everyone chasing the same thing and spending money on it is much less efficient than collaboration and dialogue where we can build very efficient research mechanisms. I think our canola value chain has got some really great examples in a variety of fronts, whether that's in disease resistance on blackleg genes or clubroot genes, genomics on things like the 50 and 60K chips. Like there's lots of good examples where industry and public researchers are really collaborating together. But I think as Dave and Wolf pointed out, it's a matter of making sure we're putting our emphasis in the right areas.
Eric, I'm gonna turn to you. You bridge this world in your role, having to liaise public and private collaboration and funding, et cetera. What are your thoughts in terms of alignment and looking for alignment and also strengthening relationships?
Yeah, because I believe, I mean, over the last probably 100 years, I mean, there's a lot of discussion about the kind of public and private alignments. And when you look at the last few rounds of clusters, and it's always results of a collaboration between the industry and the government discussions and also the researchers from the universities as well. So one question I've been thinking about for quite a few times, because I mean, we'll talk about the value chain. Like, the thing is, I mean, where is that value is being created? So right now, when you look at the canola industry, right now we're positioning canola as an oilseed crop. I mean, it's rightfully so, and it's probably going to continue that for many years to come. And do we see any other value creation? For example, right now, my understanding is that the major purpose is for oil, but the protein is as a kind of a value added. Can we somehow in the future position canola differently? What are the other opportunities? So like if we want to address those other opportunities, do we currently have the basic understanding of the fundamental science? That's where I see, like, do we have a gap in fundamental science? And also in combining that fundamental science, like a plant physiology and agronomy, with the technology available to us. Some of the researchers talk about AI, like how do we combine the both worlds, the best of the both worlds, the fundamental science and technology, and bring to the new value addition to the industry.
Yeah, perfect. Thank you. I'm gonna, in the interest of time, again, keep us moving along. I think that's really sets us up well for the next question too. And this focuses on the researchers themselves, or the HQP. So can we identify, and the infrastructure that we need to do research, can we identify any clear gaps in infrastructure or capacity in Canada to carry out canola-focused research? And how do we ensure we have enough HQP to carry out the technological research for emerging threats and technologies? Eric, I'm going to start off with you, and then I'm going to go to Wilf and pick on the rest of the panel. So Eric, yeah, if you can start us off.
Your gaps in infrastructure capacity.
You talked a little bit about even just in your last answer around, do we have the, are we aligned? Do we have the long-term versus the short-term gaps aligned? And then assuming we're working towards that, what infrastructure do you think we need to build, or we could potentially build in Canada? And how do we attract more and invest in more scientists in Canada?
You know, personally, I don't really perceive there's a huge gap there in terms of the infrastructure, but I do see that kind of I mean, with the changing climates and also different disease pressure, and also, I mean, when I look at our internal capacity within Canada with the people retiring, I mean, I want to return to my point there. We really want to make sure we have that kind of capacity in the fundamental science and to make sure that capacity stays. And so that's where I see, and not only from our side, but also from the universities and other research institutions as well.
Thank you. Wilf, I know you and I, we've talked about this before in terms of research capacity and also scalability. Like how do we scale up technologies in Canada? Do we have the infrastructure we need to scale up at times, either in the private sector or in the public sector? For some things, I think we do. Other things, I think we could maybe make some strategic investments. But I'd like to hear your thoughts around this.
You're muted right now, Wilf.
Okay, can you hear me now?
Yeah.
Okay, I'm sorry I had to switch to my phone. Something happened and I was put out of the loop on the second question. Anyways, I do think there are gaps, you know, depending on what definition you have for gaps, but certain gaps have been created, in my opinion, by the changes in the federal research system. I would just particularly use NRC as an example, was a very major player up until maybe 15 years ago, a very major player in canola innovation and research. They've almost mostly disappeared from that. That's a major loss of investment, plus facilities and research infrastructure and technology accessibility that the NRC labs provided. So I do think we need a stronger emphasis on those long-term roles of the federal labs. A second area, I think, where you know, it's a change in the way research is done. We have created, maybe gaps is the wrong word, but certainly barriers might be a better word for accessing and managing germplasm, for accessing and managing technology platforms. Those used to be relatively commonly available. For example, tissue culture, biotech tools, even training of company personnel and co-locating them and so forth. That's not done very much now in the federal labs or even in the universities. So I think there's a gap. I do think we have a technology gap in terms of welcoming and building in new SMEs. The biotech, the integration of biotech and canola was driven by SMEs that were absorbed by the larger companies. I think we need to re-examine that formula for where we want to go with canola in the long term. I'll stop there.
Dave, your thoughts?
Yeah, so I'm going to probably focus in on one area. And that is, you know, when Richard was giving an update on his economic evaluation of the impact of research on the industry and particularly the investment from the cluster funds, it occurred to me the focus is heavily on information. So if you think about the agronomic information generated with public dollars, that's pretty clean and simple, and that's just disseminated through publication, through other vehicles, and it can be used by anyone. If we think about materials and technology, it's more complicated. And so I think with so many, you know, Canada, we're reviewing everything right now. We're stepping back and saying, how could we do something differently? So I don't necessarily have the answer, but you know, I'm a bit of an SD here, so let me just throw it out. What if instead of trying to do agreements on transfer of material, and this would be under the control of public research groups, and I'm thinking specifically AAFC, what if they defined, okay, here's the project or the series of projects that leads to a certain deliverable. And when we get to that point, we're just going to release that to the industry. So we're not going to release it to one company. We're going to release it to all companies. And I think that does two things. One, it harnesses the horsepower of the whole industry, and there's still a competitive dynamic there. And once you get that in place and there's an understanding of what's in the pipe, it would eliminate some of this wastage in duplication, et cetera, that Doug was mentioning. But also, I think it'll build a different kind of relationship between public sector groups, especially AAFC, and private companies. Because ultimately, all the dollars invested in public research, the ultimate goal is to translate that into greater value, lower risk, higher productivity, first on farmers' fields, and then greatly boost value add right down the value chain. And so, that's my what I'd throw out as an area of opportunity.
Perfect. Thanks everybody for your input. Yeah, good discussion, and that's a segue into the last question then, and that is Richard talked at length, and it's a key component of this science cluster, and it's also a key component of thinking through our innovation committee as we look forward to funding new research projects, and we also look back over projects that we've all funded. What is the return on investment of research, and how can we look ahead to pick projects that can have a high return on investment, even though some of those are sometimes, as we've talked about with long-term research, they're hard to quantify immediately, but we know that there's tremendous opportunity for the industry if we move forward. And so Richard talked at length, and he has some good methodology that they've been working through. But I'm going to start with you, Chris. How do you think we can measure, high level economic impact of canola focused research?
Well, at the end of the day, Curtis, I think the most compelling measure is our industry growing in value. The research we do over time contributes to the success in the future, and so really, in many ways, those macroeconomic impact measures of an industry are really important. It's very hard, and I think Richard did a great job of lining up the complexity of trying to say, how does an individual product or an individual research project, particularly an early stage one, contribute to new economic value? Most of the tools we would traditionally use to measure are really business tools meant to say, if I put this many dollars in, how many dollars am I getting out on a profitability basis? That's a very hard measure if you're 10 or 20 or more years from a commercialized product. So, I would hope we wouldn't land on metrics that would penalize researchers from taking risks, because I think it's really important that our research community can take a swing at very impactful things. But that by its nature means that you might fail. And it would disservice to us all if we ended up with a very vanilla version of research, because it was very important that if we only just took a small step, but it was always a positive step, that would be better than taking a massive leap forward. And I think we need those big leaps forward.
Thank you. Doug, grower perspective.
Yeah, I'm going to mix my answers for question 3 and 4. But I had similar sort of comments, like just looking at SCAP. I just want to throw it out there that it can actually be for the riskier research. The government can help pay for that and incentivize taking risks, like what Chris said. Yeah, so especially for discovery research and longer term and riskier research. And yeah, if we can move back to consulting what the farmer priorities are, that will help as well. And sort of making links into Richard Gray's. So presentation. So if I was forced to say what we should do to look at economic impact, I think we can look at historical anchor points to see how far we've progressed. So if we could choose like West Star, like what are the gains that we've made as an industry compared to West Star? Like where would we be now without any canola investments? And then other anchor points could be the first hybrids in the 1990s, as well as shatter tolerance and that sort of thing. So we did, like Cody and I had like both of us had contradictory points to ourselves, I guess you could say that. So in one way, it's very hard to measure return on investment in the canola industry because there isn't one organization that's responsible for all the gains. So it's all due to farmers, commercial breeding entities, public breeders, academia, government labs. So everybody's made a strong contribution. So it's hard to say what any one single contribution has added. And then Cody also just says we've maybe become too focused on providing ROI. So yield has been stagnant for about a decade or so. So what does that say about ROA on research that has been done? But at the same time, maybe what we've done has maintained yield as opposed to we would have lost yield. So I'll just wrap it up with that.
Thank you. And Sally, I'll maybe give you the last word. You're the researcher. Any thoughts of how you would like, or you view research and valuation of research? And I noticed Richard has his hand up, so I'll maybe pull him in for a second too. But
okay. Yeah, no, and it is really good to hear multiple voices. Chris most recently kind of proposing this holistic approach of looking at the industry as a whole, and understanding that it is really tricky to break down the contribution of a trait from an ROI perspective, especially when it is quite risky to look at. One thing we're contending with within the canola industry, I'm going to quote Guelph from about 10 years ago. I remember him making this comment in a similar discussion, in that in the canola industry, from a public researcher perspective, we're in a bit of a tricky situation, because when it comes to publications, it's hard to know how much industry is also looking at those publications. As we're presenting at forums such as this and Canola Week, how much of an impact is those public disclosure of results and the methods that we're using impacting the industry? And that's where it gets really, really tricky to measure, because we don't have an impact factor like we do with scientific publications. So I think, knowing that there's understanding across the industry and the experts that are advising in terms of making science decisions, that's really good to hear. Yeah, maybe I can leave it at there. If you wanted to pull, sounded like Richard might have some comments that would contribute as well.
Richard, if you might have a comment, and that gives me, if you go quickly, get 2 minutes for closing remarks, and we're out of
Okay, sure. I really appreciate these comments. I think our best shot is looking backwards and looking at disease resistance that has been added to canola from some of the GF1, GF2. I think those types of things are probably large. If any of that genetics has got to the commercial pipeline, I think you'd probably know that from your genetic work. Am I wrong in saying that you can measure that some of those genes have been implemented? Is that correct?
Yeah.
So that would be one place I would certainly want to look at. And certainly, I know 30 years ago, there wasn't a lot of interest in canola meal generally, but there's a lot more interest in canola meal in feeding. And I think that's due to the research as well. So some of those pieces, I think, that can be quantified. So I'm going to leave it at that, but I certainly welcome some input from everyone if they've got that.
Perfect. Thank you very much to the panelists. I really appreciate your input. I know we're going to continue this probably in some way, shape, or form at our various venues in the year to come. And so look forward to that as well. So thank you very much. And with that, I'll just close out the day quickly with a few remarks and thank yous here. So we've had a great midterm overview and checkup on our key sustainability Canadian Agricultural Partnership Science Cluster projects. These projects were all vetted as high priority by the canola growers and the canola value chain members who fund this valuable research. And these projects fit the AAFC priorities of greenhouse gas reduction and carbon sequestration, economic growth and development, and sector resilience. And they're also really integral and incredibly important for our CCC innovation pillars of performance, precision, protection, and product. So we've heard a lot of preliminary results for key activities, including disease and insect management, climate adaptation and resilience, nitrogen utilization and optimization, plant growth and yield optimization, meal utilization, and economic return on research investment. And equally important is the translation of research into best management practices and extending and transferring these best management practices to agronomists, practitioners, and producers to realize the innovation that is generated. So we've got a good reflection on that in terms of some of these important tools that we use to translate knowledge into practice. All of these serve to increase yield, increase profitability, reduce production risk, and improve our environmental footprint. In closing, I'd like to thank all of the presenters today, and a sincere thank you to all of the researchers on these research teams and projects. These researchers don't always get to present, but they work so diligently on these important projects, and without them, we wouldn't have this opportunity to dissect, discuss, and analyze and interpret research for the benefits of our industry. I'd also like to thank my colleagues at the CCC for organizing and participating today and for the work they do on the knowledge and tech transfer front. On behalf of the canola value chain, so Taryn, Chris, Jay, Caroline, Wade, Warren, Keith, and the entire crop production innovation team. I'd like to also thank Eric, Liu, and AAFC staff for helping to pull this day together. It's been a pleasure, as always, to collaborate on events like this that advance the knowledge and the research of this particular science cluster. I'd like to thank Chris and Haley for being excellent chairs today, keeping everybody on time, being engaged, and bringing together some excellent panelists as well as for questions. A real special thank you to Ellen McNabb at the Canola Council of Canada for competently and effectively and efficiently assisting with the administration of the cluster research program, both to our researchers and also our own crop production innovation team, and also to Tammy Campbell, who is our program officer at AAFC. And finally, a big thanks to all of our research funders, our core funders at the Canola Council of Canada, our provincial grower organizations, Alberta Canola, Sask Oilseeds, and Manitoba Canola Growers, and to Agriculture and Agri-Food Canada, our public partner in this strategic alignment and research. They make this valuable research possible. Thanks again to everybody. Enjoy your summer.